You can see here this pathogen has a very rapid evolution. But this can be seen with the cloud number of gene is more than 11,000. You can see these two new species are clearly forming groups here, independent group. With this species, we also confirm their ANI and DDH, and we can clearly see that the ANI, when we say new species, is supposed to be less than 95%, while the DDH is supposed to be less than 70%. And you can see these two both qualify for it. It's less than 95%, it's less than 70% here. This is the most up-to-date taxonomy of this species. This preprint is available, but the publication is still under review. So this is where we start the study. We screen hundreds of strains for virulence and pathogenicity. One thing that we found very interesting in one of the strain that was not producing EPS, but is still highly pathogenic. So what we did, actually, we selected then 3 strains. Generally in this species, it happened that when it's not producing EPS, it's generally non-pathogenic. So we picked this strain that is producing EPS, non-producing EPS, both virulent, and then we pick one that is producing EPS and non-pathogenic. What we found interesting here, but before that, let me explain the experiment. What we did here is we took 3-week-old tomato plant, inoculated them, and we harvested tissue after 30 days, 1 centimeter above the inoculation point, and we then did the electron microscopy. What we found here, you see in the greenhouse, we see no symptoms with when we inoculated with butter and when we inoculated with the non-EPS producing. And you can see over here, no symptoms, no canker, no collapse of xylem, and we can see the strain is EPS producing. While when we have the virulent strain that is producing EPS, you can see building symptoms, and there was some clear canker, collapse of xylem vessels, and you can see over here is EPS producing. Similar story with EPS non-producing, but highly virulent, canker, collapse, tissue collapse, and then you can see there is no EPS production on this one. When we had those stem section, transversal and longitudinal, you can see very clear vessels here, and same story here with non-virulent EPS producing strain as well as with the PBS inoculated plant. While when we see those pictures, this is completely collapsed in case of virulent and EPS producing. Virulent, non-EPS producing, the story is the same, complete collapse of xylem vessels, and here both cases as well. When we zoom the picture, you can see the vessels are very clean, no sign of any bacteria in the EPS producing non-virulent as well as the PBS. There is no sign of bacteria. While when we have this virulent EPS producing, you can see bacterial growth here, but not completely chalked. And you would see these symptoms later on. I would have a picture showing that is matching with what we are seeing here. While with the EPS non-producing virulent strain is completely chalked on those vessels. And as I said, this is completely matching what I would show you later on, why this EPS non-producing infected plant has really sudden death of the plant. We also check the phenotypes of these bacterial cells, because these were EPS producing, non-producing, to just see if there is any correlation with the phenotype and the pathogenicity. We didn't find any differences. So we took hundreds of pictures, but all saying the same story. Then in the next experiment, what we did, we actually inoculated plants with these all treatment, I would say 3 strain and 1 control in different replicates. And you can see these are the inoculation point. This is the inoculation point, and these are the point where we harvested the tissue after 10 days, 20 days, and 30 days. And then we quantify it through the duplex Stackman qPCR assay. What we found here is very interesting, that when we harvested tissue 10 days, we are seeing some of the positive sign for non-virulent strain in root, while this is not present after 20 days and 30 days. So it was only over here, and we see the gradual increase. So you can see here, we see the high growth compared to 50%. We only see the EPS producing. That's what exactly is saying. After 20 days, we see the gradual increase in the in colonization of EPS producing pathogenic, while there is no EPS producing strain now detected. While we are also seeing that EPS not producing clay vector is also gradually going, but not with the same speed. But after 30 days, we see a sudden increase in the colonization of EPS non-producing virulent strain, and that's exactly what we have seen in electron microscopy in previous slides, and that's what we have seen in the letter as well in other experiments. After that, we try to understand the virulence mechanism and virulence determinants within these clay vector, and we see there are three important part, and one is the pathogenicity island that include the CHP and PP and TOM clusters, and then others are the PCM1 and PCM2 plasmid to see their impact or they're really important for pathogenicity. This is just indicating what the cluster involved. Here in the experiment that was done by Gartman et al. In 2008, they did an experiment, create mutants. What the first thing that they are showing, when they have no mutant, like they have plasmid and PAI present, you can see the wilting and symptoms over there. While when they remove the plasmid, there is no wilting. While then remove only the PAI, no wilting, and control no wilting. So it's indicate with this, they concluded with this experiment that the plasmid and PAI both required for the pathogenicity. But this is just about the exopolysaccharide, because we had initial hypothesis that is playing some role in pathogenicity, but we didn't find so far any correlation with that. Here, what we did, we sequenced about 3 strains, and what we found interesting here, you see that PCM1, so this is a reference strain that we compare with, that they also use in the previous study, and we compare, you can see they have PCM1 plasmid, PCM2 plasmid, but when we check our strain that is EPS producing virulent, it did not have this PCM1 plasmid. That's indicate that PCM1 is not required for pathogenicity. Similarly, the EPS non-producing indicate it has the PCM1, but only 35% similarity, but do not have PCM2. So it's indicate neither the PCM1 nor the PCM2 is required for pathogenicity.
Okay, yeah. Actually, some glitch, maybe connectivity with Oklahoma or something. And without taking delay, we invite our second speaker, Dr. Patwa Singh. She is Regional Manager, South Asia for Australian Center for International Agriculture Research and based at Australian High Commission, New Delhi. And she is going to speak on ACIR research experience on climate resilient agri-food system in South Asia with focus on plant disease management. So I welcome Dr. Patwa Singh for this inaugural session of panel lecture.
Yeah, it seems like in the morning there is a lot of technical issues. And I have been told that the previous presentation is also recorded. So maybe it's a good thing, so we can access later. So just coming back to my presentation, first of all, I like to thank Amity organizers, ISMPP, on behalf of ACIR and also on my personal behalf. This is my first time visiting Amity. And above all, I just like to thank Dr. Shalini for inviting me. So I'll just, this is outline of my talk today, like what I'm going to talk today. So number one is what we do and how we do in terms of organization, and short overview of investments and impacts. Then ACR's current research projects in South Asia. Then coming back to plant disease management, there are three key findings, which I will be briefly sharing. So first is mitigating effects of stripe rust on wheat production in South Asia and Eastern Africa. It's by Dr. Robert Park, University of Sydney. Then International Mung Bean Improvement Network, Developing Disease and Pest-Resistant Varieties, by Dr. Ram Nair, World Vegetable Center, and Managing Wheat Blast Diseases in Bangladesh by Dr. Pawan Singh Simit. So my presentation will not be very technical. I will just try to present what are the findings of these projects. So as part of Australia's foreign aid program, ACR funds connects agriculture researchers in Australia to developing world, achieving more productive, resilient and sustainable agriculture. Basically what we do, we fund, facilitate and manage a research partnership in agriculture research. Then this is just operating in 36 different countries with around 385 partners in Indo-Pacific region, managed by 11 country and regional offices. So one of our in New Delhi, which is a regional office for South Asia. There is another office, country office in Pakistan, which manages Pakistan. So our office manages Bangladesh, India, Nepal, Bhutan, Sri Lanka. Earlier, we used to also manage Afghanistan, but due to the difficulties, we were not able to. And then overall aim is boost agriculture productivity, sustainability, and food system resilience. Our focus is natural resource management and climate change. We place high priority on evaluating impacts and learning lessons, capacity building at various levels. So ACR also funds, at the moment, we have around 100 ACI alumni across South Asia. We have like fellowships for masters, PhD, then leadership fellowships, and specifically one Mary William Fellowship is specifically focusing women leadership. And then we are, as obvious, we are part of Australia's international aid policy. Our focus is climate. 90% of our project investment should be climate. Gender is another focus, meaningful gender inclusion at all levels, and locally led. So it's not that it's Australian agency working for Australia. It's the investment what we do, the issues has to be from nationals. So like for it is working for India's national priority or Bangladesh national priority, something like it's not working for Australia. So yeah, so now the question is how we do. So basically, it's a very simple thing. It's like mainly we do evidence generation through locally appropriate innovations and research partnerships, capacity development, which I already mentioned, then partnerships and platforms that enable adoption and scaling, gender equality through international gender responsive agriculture research for development. So with the partnership and platform, which I mean like all ingredients are there. For example, in Indian system, it's the largest, ICR is the largest agriculture research system in the world. So what Australian partnership is adding? Number one, it is adding leadership and convening and convergence of different platforms. So during my presentation, you will have idea like how we enable these. Then another important point is long-term partnership, tracking adoption and evaluating impacts are particularly important. So these are the like main drivers for, and So Asia doesn't work only in South Asia. We have Pacific, Southeast Asia, and Africa. And in case of South Asia, at the moment, we have 27 projects with 13% of investment. So this is just an overview of our investments. Then, as I mentioned, tracking and evaluating impact is another important. So recently, with the Indian Council of Agriculture Research, we completed, actually not recently, 2 years back, 40 years of research partnership with Indian Council of Agriculture Research. And there are 2 publications which has like overall benefit. So if you can say the benefit, average benefit cost ratio for return on investment is 42:1. And the next slide is just, next figure is just on like what are the impacts pathways, and also what are the with the research areas. So here you can say with the pest and disease impact pathway, there are 13.1 billion across our project portfolio. So these are the regional drivers, why we like to work regionally. There is a shared pressure on land, water, natural resources, climate variability and falling water tables require regional-wide solution. Then diversification beyond cereal is growing priority. And our focus is a resilient system across highlands, plains, coastal areas. Emphasis on gender inclusion, climate smart agriculture, and policy reform. Here I can also add another C is conflicts, which is also affecting another driver to work together. So these are just a list of some of the ACR project, International Mung Bean Improvement Network, Bangladesh, India. It's mainly improving varieties, it's a regional collaboration, multi-country collaboration, managing wheat blast in Bangladesh, hybrid wheat breeding, additive intercropping with Bangladesh, India, and also we recently included Bhutan in this project. Which is a basically diversified cropping for climate resilience. So these are our cropping system, farming crop and farming system research portfolio in South Asia. Then there is a water, climate, and natural resource management. One is transforming a smallholder food system. It is all ongoing project with in West Bengal with UBKB and in Bihar with Bangladesh, Bihar Agriculture University. Then spring water management for resilient livelihoods. It's a new project we just started in Nepal, which is resource security and inclusive rural development. Then there is another one with the forestry program, Bangladesh Sundarban ecosystem management, cropping system intensification in salt-affected coastal zones, Bangladesh and West Bengal, India. It's basically improving the land productivity, profitability, and resilience. So in this picture, you can see that what I was talking about, the converging different platform. The person who is in the field is agronomist from UBKB, Uttar Banga Vishwavidyalaya. And you can see the back is West Bengal state government agent, Department of Agriculture is there. Then there are all farmers. So instead of working in isolation, we involve all the actors on platform, including the farmers. So coming back to disease management, I just try to focus. There is different natural resource management, but particularly since it's a International Society for Mycology and Plant Pathology, so I pulled all disease management project what we are doing in South Asia. So here you can say, I don't need to, the people here in expert, like why these wheat disease it is important. Like there are 3 rust diseases among leaf rust, stem rust, stripe rust. And if you can see the figure, it's enough to feed what annual losses we are having, enough to feed 35 million people. So it's emphasizing again why disease management is important. Then this was the project already closed in July 2016. June 2020, it was a 4 years project investment. And there were different partners, University of Sydney, then New South Wales, Department of Primary Industry, ICR, IIWBR, Indian Institute of Wheat and Barley Research, then Pakistan Agriculture Research Council, Nepal Agriculture Research Council, and here Ethiopian Institute of So it was basically a multi-country project, and this was one of the maximum return on investment project which Australia invested. And basically it has like a different characterization, resistant to yellow rust, then also validating different genes, undertaking stripe rust pathogen surveillance, build partner capacity in rust pathology and genetics. And just I will not go into technical details, but this is basically, this project provided different rust-resistant donor germplasm and marker linked to important resistance to breeders. So this is just like how many wheat lines were, like 3,000 wheat lines were screened all across different countries, including Australia. So, and then there were some surveillance also, which I'm not going to, because pathogen surveillance work is also very important and highly linked with these findings. Then coming back to International Mung Bean Improvement Network, which is a kind of a unique and important network for mung bean improvement in the world. And actually, personally, I knew in Mung Bean International Network before joining ICIR. So ACIR, because this is so important network. And the reason is that CGIR doesn't have a mandate crop for mung bean. Only World Vegetable Center has mung bean. So although we are core funder for CG Institute, but we have also included mung bean as an important crop. And actually, these slides are shared by Dr. Ram Nair, who is World Veg Center from Hyderabad. I will not go into the causal organism. Everyone is here in this gathering are familiar with the mung bean yellow mosaic disease, and which is a very important disease. In the figure, you can see that it just totally lost all the mung beans. And this the graph chart above is just highlights, it's maybe not visible, but our all details are present in the website. But just to emphasize that although it's a multi-country project, but every country has their own priorities. So someone has bruchid resistant, like a storage pest. Someone has a yellow mosaic. So yellow mosaic was most of the people were interested in yellow mosaic disease resistance. So just the outcome, this is OUAT also released with few resistant varieties. So these are the field performance of OUT Kalinga green gram. And you can see this pictures. Then another one is with the, we transferred one to Daftari seeds. And they also really, this variety, Daftari Gaurav, is a high yielding, it's a project, it's like the seeds were transferred to the company, and they have started marketing in the state of Maharashtra, Madhya Pradesh, Rajasthan during spring season. So then another disease was like dry root rot, which was taken care in mung bean. So this work is still ongoing, but there are some important lines have been identified, and some NBS LRR proteins are involved in this, which is still undergoing research. The project is still ongoing. Then coming back to Wheat Blast project in Bangladesh, I'll not go into the causal organism. It's a Magnapurti rhizae pathotype triticum, and wheat blast is a seed-borne airborne disease, and resistant sources identified but limited knowledge. And this population is, the pathogen population is very diverse. So I had experience working with the rice blast, but it's a similar kind, like it can be virulent depending on the resistance gene, and can be attacked any aerial parts of wheat plant. So this is just to give you the intra and intercontinental spread of wheat blast. So you can see these are different countries, part of, and in 2016 it reached to Bangladesh. So it's basically, it's because of grain trade, it is spreading. And when you see the wheat blast reaching to Bangladesh, it's very adjacent to Indian wheat growing region. So although the air is in our favor, like it's not coming to Indian side, like mainly air is going from India to Bangladesh. So in terms of spread, but still we need to be cautious about this. So this is again just emphasizing climate change. So in favorable conditions, this at the mid wheat blast alone could reduce global wheat production by 13% due to favorable conditions. So just wanted to mention this. And this work is done by CIMMYT, Mexico. Then this is my final slide. I just wanted to highlight why international collaborations are important. So when you see that wheat blast coming in Bangladesh, it was migrated in 2016, and it was Bangladesh scientists observed a resistant CIMMYT breeding line in NVR trial. Then wheat blast resistant confirmed by USDA ARS and also fields of Bolivia. So it was like quickly identified and screened, and resistant variety Bari Gom 33 was released in 2017. So maybe it is the fastest release of the, it's not because, it's because of international collaboration, because we were able to tap of all the knowledge available in this area. And just to give importance that there was a wheat burning in West Bengal farmers because of this disease. And these are all the vulnerable areas in the Indian map for this. And projected losses were 1.32 billion USD in epidemic years. So yeah, so here I like to stop. And if any questions, thank you.
Thank you, Madam. We thank Dr. Pratibha Singh for his insightful lecture and all research going on around India. Anybody have any questions, he or she can ask. Thank you,
Otherwise, I am available online as well with my email. So.
there is no question.
Thank you.
Now we invite Dr. Shiv Kumar Agarwal, Director FLRP India, ICARDA Morocco. He will be speaking on impact of climate change on the disease spectrum of cool season food legumes and their management. Dr. Shiv Kumar Agarwal, please.
Thank you very much. So good morning, everyone. At the outset, I would like to express my sincere thanks to the organizer for inviting me to make a presentation among the pathologists. I am a plant breeder. So on behalf of Neha Tiwari, she is our pathologist. I will be making a presentation, and these slides are prepared by her, by the way. So I'm going to make a presentation on how this climate change impacts our disease spectrum in legumes. As you know that ICARDA is working on a number of legumes, mainly food, cool season food legumes. And if you see that the question comes why we are working on legumes. As you know, the legumes are very important crops in sustainable agriculture. They are good for the people as well as good for the planet. They are good for the human health because they are having more proteins. They are also having a more micronutrients. almost 2 to 3 times more protein than what you see in cereals. And when you have legumes with cereals, I think that is what you get the balanced diet and the biological value of the proteins get enhanced. At the same times, they are also good for the soil health. Why they are good for the soil health? That they fix the atmosphere nitrogen. At the same times, they also make the rhizosphere so suitable for bio agents that many other nutrients like phosphorus also becomes available for the plants. And of course, they are the very integral part of any strategy where we want to diversify our cropping systems. As you all know, that monocropping of cereals has been affecting our soils, and it is also having an impact on our water availability. It is also affecting our nutrient status of the soils. So if we have those legumes included in the cropping systems, then the system productivity get enhanced. The whole landscapes change. And if you just see that, legumes, then they are being grown in as many as 52 countries. And at present, they are being, grown in around 95 million hectare lands, producing 96 million tons of protein-rich grains. If I take into account in India, then we are producing 24 million tons from around 26 million hectare land. But then, if you see the last 5 years data set, the area under legumes is going down, production is also going down. And then, at the same time, we have to have around 32 million tons by 2030 to ensure that we are going to bridge the gap between demand and supply. It means, and on top of that, what happens? On top of that, if I take this climate change, the overall vulnerability of crop productions, then you'll find that our global south is going to be impacted much more than what you see in global south, the global north. And under those conditions, when the climate change is there, it is not that the direct impact is there, but also the your disease spectrum is being changed. And that is where we have to think in terms of how we should design our strategy to manage the disease so that we are able to fill this demand supply gap of pulses. If you see in legumes, the major diseases, some of them are foliar diseases like ascochyta blight, botrytis gray mold, and then rust in lentils, lentil and pea. So number of foliar diseases are there. At the same time, you will see that pulses are mostly affected by the root diseases. And this root diseases are a kind of complex. You have fusarium, you have a collar rot, you have a dry root rot, number of diseases are there. If I combine, and of course, then with the climate change, now viral diseases are also become very, very important, particularly when the temperature is rising. If I take all those diseases into account, on an average, almost 22% yield losses are there. But of course, some of the disease, sometimes you can get 100% losses. And then if I calculate globally, what are the economic losses? It is coming to around 220 billion US dollar annually. So that shows the kind of gravity of the situation, that if we are able to manage our diseases, this much losses we can. And when we say that we want to increase our pulse production from 24 to 32 million tons, if with this 22% losses are managed, then we will be able to meet that, we will be able to bridge this demand supply gap. But then with climate change, it has impact on this spectrum. And these are some of those factors which affects most. If I say that the high, this high humidity, this is the one factor, almost, it contributes around 37% to changing the, disease spectrum. followed by high temperature. And of course, there are other, like extreme weather as well as low temperature. They also contribute towards this, to change the disease spectrum. And of course, this, all these factors, they impact the pathogen distributions. They impact the host susceptibility. They also, impact on the pathogen survival. Besides, of course, when it comes to the viral diseases, it is the vector population that is also being, affected, generally increasing, the vector populations. And of course, with the change in climate, the diseases are changing. At the same time, within the disease, the whole, racial picture is also changing. And many of the races which were very prominent earlier, particularly in Tunisia, earlier it was race 0 of fusarium, which was more important. Now with the climate change, now it is becoming other. Now race 6 and 7 has appeared. So number of races are also appearing. And then it is not that all climate factors affect the crop equally. Different factors affect different crops differently. For example, if I take into account the fusion wilt, it is about the temperature. Temperature is the most prominent factors, climatic factors, which impact the fusion wilt, intensity and severity. And when it comes to botrytis grumult, it is about the moisture and light. Similarly, for different diseases, it is a different factors which impact that one. In this one slide, which I want to say that how this change in fusion wilt distribution under the climate change. If you see that it's globally distributed, but it is more in the particularly global south, sub-Saharan Africa as well as in South Asia. But then, in future, with the change in the temperature, it is expected that this disease will be expanding toward the global north. Means it will be impacting our European countries and Canada, all these things, even our North Indian conditions. If you see today, in North India, in chickpea, it is ascocarp blight, which is more prominent, and that's how the area under chickpea has gone down. But the change in temperature, it is expected now this fusion wilt will be more prominent. It is the root disease, we just wanted to show in this slide how this temporal as well as spatial dynamics of fungal pathogens are associated with the chickpea wilt, complex. And then you see this complex, it is, at different growth stages, then different kind of, pathogens are there. If you see initially, it is mainly about the color rot. When it goes after flowering, then it becomes dry root rot proportion keeps on increasing. So that's how it keeps on changing as per the crop growth. So therefore, when we think in terms of developing the management strategy, we should also think in terms of how we should go for this growth stage. At what growth stage, what kind of strategy we should apply. And then that this is one example of lentil color rot. Earlier, if you see that it was not a very prominent disease, but with climate change and with more moisture, now this is becoming very, very serious. So now fusion wilt is under control, but now color rot is one where we want to do, and for that we have collected different isolates from different places in India. And we have done this phylogenetic analysis, among this Sclerotium isolates. And you can see that then we found that all those isolates, mostly 90%, were, from the Sclerotium rhodophyll. And then among the foliar diseases in legumes, of course, is ascocarp blight, which is very, very important. Of course, in lentil and pea, it is rust, very, very important. Similarly, for fowlbean, it is chocolate spot. It's very, very important. And what we did, under that, we have done this, kind of, genetic analysis, and we could see that this particular ascocarp blight in chickpea, it is controlled by a number of genes. And it has a different kind of, genetic regions, which we have now tagged, and now they are Now we are using our breeding strategy where we can deploy these genes through the speed breeding. And of course, within speed breeding also, we are implementing now how we can screen our segregating populations. So that is what we are doing in that. And this is one of that. Nowadays, as you know, that now AI has become very, very important, where we can think of predicting this AB resistance in chickpea. So what we have done at these three different locations. Of course, this is a GRD supported project under the three different locations: Ethiopia, India, Lebanon, as well as Ethiopia. We have collected those exciter isolates, and we call it pathotypes. And then we have evaluated them at different environments. And based on this, AI, we were able to, without even scheming, we are able to identify the resistant lines. That is the kind of, advantage having AI when you integrate into our programs. Then we can predict AB resistance in our chickpea lines without even testing them in the field conditions. Now, as I say that, with climate change, particularly change in the temperature, rising temperature, now viral spectrum is changing very fast. Some of the viruses which are becoming, just now Pitwa has mentioned about in moonbeam, now yellow mosaic virus is a very devastating disease. And I remember earlier, we had no solutions, but now we have a number of variety which has come from different projects that they are doing very well. But then in case of lentil and particularly cool season food legumes, these are some of viruses which are very, very prominent, like wheat western yellow mosaic virus, chickpea chlorotic resistant virus, bean leaf rolls virus. Then we have this following necrotic yellow viruses and soybean dwarf viruses. These are the ones which are happening. And when we import our material from Morocco and Lebanon, and we are planting in India, then these viruses become very, very important, because the temperature here is higher than what you get in Morocco and Lebanon. And then we have gone for this field screening. Then we have this, the PCR based, primers for screening. And we were able to identify the resistant sources for lentil as well as chickpea for different viruses. And then many times, just because of the change in the climate, you will find that the vector dynamics changes. And because of that, what happens that, and they are the one which really play a major role in transmitting the virus diseases in the legume crops. And you can see that, this temperature with the change, what it does, it expands the geographical range of the viruses. It increases, extend the breeding, seasons of the virus as well as then of vector, and then increasing the survival rates. And all these things contribute to the higher virus, incidence in our crops. Then all those diseases, if we have to think in controlling, I think we have to think in having an integrated approach. And for that, of course, we have to think in terms of monitoring and survival, and then host plant resistance, followed by biological control. And these are some of the practices which we have to integrate together to manage diseases. Because it's not only about having resistant varieties. When you have the resistant variety in the central, and then you have all other options available, then we are able to manage them. And once we do that, I'm sure that we can be able to meet this global demand of pulses. And then if the future research priority means, then we have to think in terms of climate disease modeling. We have to think in terms of genomic-based breeding. Then we have to also think in terms of advanced diagnostic. And of course, by using the AI tools, we will be able to screen our germplasm. We will be able to predict our, we will be able to forecast also about the disease, intensity and survival. So with this, I would like to express my sincere thanks. Thank you very much. For 10 minutes, I think that's what we can complete. Thank you.
Thank you, sir, for your insightful talk on the effect of climate change on the evolution of disease, and particularly the important food legumes. If anybody is having any query, he or she can ask. Thank you, sir. There is no any If anybody is having any, he can directly talk to him. Now we invite Dr. Alam Suresh, plant pathology lead, CIMAT, Sub-Saharan African, nearby Kenya.
Can you hear me? Can you hear me?
It is a pre-recorded talk.
No, can you hear me?
Yes, we hear you. Yes, sir. Yes, sir, we hear you.
Okay. Just give me a second. I'm just switching to the, yeah. 1 second.
Yes, you can start. We are hearing you.
Yeah, just a second. Can all of you see my screen? Can all of you can see my screen?
Yes, we can see. Yeah,
Okay.
please go ahead.
Yeah, thank you so much for giving me opportunity. I thank organizers for giving me this important opportunity to share one of the most important work in Sub-Saharan Africa. I fully echo the previous speaker and from ICARDA and also Dr. Neha Tiwari. She has done a wonderful work, and also the concurrent sessions. It has been very nice. So first of all, a good morning to all of you. Most importantly, the maize lethal necrosis is one of the most devastating diseases in Africa. And especially, it is not just a disease, it is a lifeline, it is a livelihood, it's an income. It's more than that, it is economic foundation for several million farmers in Africa. In Africa alone, around 35 million hectares of maize is being grown, and almost 70 million tons. So it's average of 2 tons per hectares. So it has been affected by several fungal diseases and ear rot and viral diseases. Among viral diseases, maize lethal necrosis is the key viral diseases. So in this viral diseases, just to give a perspective, maize is affecting almost 22% of the crop loss in the world, and in which Africa alone is just having 1.5 tons per hectare as compared to 5.5 tons per hectare in the globally. And to give you a perspective that this particular viral diseases is not one viral disease, it is it's combination of two viral pathogens, especially maize chlorotic mottle virus and sugarcane mosaic virus. When they combine together, they cause a disease called maize lethal necrosis. It is reported in Eastern Africa in 2011. Since then, it has spread to all the Eastern African countries like Uganda, Tanzania, Ethiopia, Rwanda, DR Congo, and all the neighboring countries. So this particular disease is further not reported since 2016. But in 2014-13, the Dr. Hugo has done an extensive study to understand the economic implications. It is almost $185 million per year in Kenya alone, having the losses of the disease due to the loss of crop. So why this particular disease in Africa? It can also come to India. There is no way that it cannot come. If it comes to India, it is more havoc. But MCMV, maize chlorotic mottle virus, is new to the region. Potentially could be new strains of potty virus, or sugarcane mosaic virus, or dwarf mosaic virus. In African environment, it is very conducive to grow maize because of bimodal irrigation, so rainfall is condition, and leading to continuous buildup of virus inoculum. And you all recognize that it is mainly because of seed contaminations, and it has affected the whole our regions, and it's also affecting the seed trade, making seed trade disruptions. And widespread of susceptible germplasms. As we saw and studied, more than 90% or 99% of the commercial varieties even now is completely susceptible for the diseases. So this is where the disease is very vulnerable. And if you consider because of this the crop loss and economic seed trade disruptions, income loss, and CIMMYT together with partners, they have worked together closely in to demonstrate science and collaborations. More than that, how a teamwork can be played a significant role to combat a major threat to African conditions. This is one of the key examples. And basically, this particular disease is being transmitted by seed. Yeah, especially it is transmitted somewhere in Gluma. Please remember why I'm coming to highlight this one. I will have another important point to highlight in the next slide, next coming slides. And it is also transmitted by various vectors, instruments, crop debris, seed, and so many ways it is transmitted. That's why it becomes more challenging. So, and we did not just put the points as a PowerPoint, but we made the journey in each of those honeycomb very seriously last 11 years with 8 to 9 countries and more than 25 partners. First and foremost is the breeding for deployment of resistant varieties, which any disease management strategy taking place. Rigorous monitoring and surveillance, which I will discuss with later, with close collaboration with partnership with the NPPOs, and diagnostics and epidemiological studies is detailed has taken place. Seed-free, disease-free seed productions and exchange. This is another most important area which we worked. And agronomic management practices we trained, and capacity building. Around more than 9,000 people around the regions has been trained. Around 400 inspectors in 8 countries, 10 years has been trained, including extension workers. And they're all making a champions to combat this disease. As I come to the first point, that the most important element here is the developing disease-resistant varieties. And CIMMYT, in close collaboration with the CALRO, like ICR in India, they have established 20-hectare facility to develop, identify, and develop and deploy disease-resistant varieties. This is fully quarantined and having high phytosanitary standards in terms of inoculation protocols, in terms of facilities, in terms of process, including for automated fertigation scheme, and all the phytosanitary measures has put in place. And you can see this is one of the leading commercial hybrids, even now, is completely devastated by these diseases. CIMMYT, in response to this, CIMMYT has given a very good response to it. So most importantly, I'm coming to the disease surveillance and diagnostics. In some of the areas where diagnostics should be very simple, effective, and implementable, CIMMYT has optimized, we have optimized the diagnostic protocol and using simple ODK tool platforms and immunostrips. And with these digital tools and technologies, we are able to have this surveillance in entire 8 countries in 10 years. Almost 19,000 disease data points has been established. So this is one of the hallmark for entire maize in the world of disease surveillance. And if imagine it is like an entire, looks like India, but it is entire 8 countries been thoroughly tracked. Even now, almost 20-25% disease is still prevailing. And we can easily know, this is an example how Rwanda has taken seriously on their own sustainable funding without any external funding. And this is how they take it very seriously with little technical backstopping. And you can see how every year they're being monitored and last 10 years. And then one most important aspect is that we developed another point of the most important is that we developed a seed health testing, especially in Kenya, and also in case of Harare, Zimbabwe, a stringent quarantine facility has been established. And recently, we have developed a seed treatment methodology, first time in the world, and designed by me. And the protocol is very new, and it completely decontaminates the viruses. As my previous speaker has said, resistant variety or hybrids alone is not sufficient. We have to combine with several methods, thereby we can combat the diseases in a most sustainable way. And more than that one, last 10 years, we have shipped more than 100,000 or more than a lakh germplasms to the entire world. Not a single seed has been affected by viruses. Imagine one seed has come to India, Kenya, because of seed movement. But we have shipped more than 1 lakh germplasm, not a single seed has been affected by diseases. And it is well certified by regulatory authorities. And not just that one, recently we have developed a seed tracker digital tool where we can easily track, trace, and treat the seed where it is being produced, which aspect has been affected, and all the management practices monitored. All those things are under observations. So, and more than that one, we constantly train all the seed companies, NPPOs, and NAS for how to produce disease-free seeds using the checklist established by in consensus with various partners. And this training, I can say more than 6,000 people have been trained in entire Eastern Africa. Yeah. And as previous speaker also mentioned, the climate change, we also see how futuristic 2030, 2040, how futuristic climate will affect these diseases. And more likely, it is still remaining in Eastern Africa. But if the disease hits in Southern Africa, it becomes the most important challenges in the coming days. So, but we have put in very stringent measures. However, we also studied a transboundary quarantine measures. You can see entire Africa is having a porous transboundary mechanisms. Even I can say, even India, between the states, there is not much significant checks and balance to combat any movement of plant materials. That's why some diseases starting one state, you can easily go to another states. But if you see here, all their airports and testing facility, border points, quarantine facility, they have been thoroughly studied last 2 years. It has been found that this entire ports and bars are very vulnerable. That's why they need to be strengthened in capacity building. Most importantly, this is the hallmark of any breeding program. When I started, at that time, there was no resistance source. At that point, at least we identified some resistance source, bringing one source. And 2016, we deployed almost 5 tolerant varieties, whatever it is available. Since then, we focus more on the targeted resistance breeding program. And then we further enhance the capabilities by continuously improving the resistance hybrids. Like this, we have deployed and released 20 hybrids to the partners. And that is one step. Second, developing molecular markers. And then third is that developing MABC platform, marker-assisted backcrossing. So this particular line, for example, is very widely used in entire Africa for the last 3 decades. It is very good for drought. It is very good for other traits, but it's highly susceptible for maize lethal necrosis. If you see here, using the simple molecular-assisted backcrossing, the same line has been converted to resistant source. This is another key areas. More than 100 lines has been like this has been converted. When we started, the average mean of this resistance is almost 7.5. With the continuous change in the breeding strategy, use of novel tools and technologies like double haploid technologies, molecular markers, the mean has shifted from where it was 7.5. Today, I can witness almost 2. Here is the key change in the breeding program. And not just that one, our quality of data is more than 95% heritabilities, which is consistently established over several years. And through these 12 years, we have supported 12 NARs and 1 NPPOs, 17 private companies, more than 200, now it is 280,000 germplasm has been screened, more than 350 lines has been identified. Now you can easily see gene editing technology, first time in Africa and also world for this disease has been developed and deployed. Now it is ready to make a new hybrid combinations and release to the farmers. And definitely you will be thrilled to see by yourself. There are several Indian colleagues have visited this experiment. And it has been published in Plant Biotechnology. And last week, it has been accepted in PNAS 2026 journals. And now why I'm putting this particular the picture is that if you see here, all the resistant hybrids having 6 to 8 tons, whereas susceptible, no yield, 0 yield. And then tolerant is almost 4 tons per hectares. And this is how we make very significant progress in the breeding programs. And these all lines and hybrids available to the partners in the entire Africa or in the world. It can be easily available. Yeah, just last two slides.
Yeah, sir, please.
Yeah.
Yeah.
And as I mentioned that more than several capacity building, first most important emphasis is that this more than 9,000 people in the region have been trained. So developing technology is one, and consistently making them, enabling them to use and adopt is another one. So many people have been trained in terms of different areas, and this is one of the key areas of CIMIT, and in various aspects of disease diagnosis and surveillance. So in the final slide, this is the global model or a fingerprint, like a future-proof model from a regional success to global model by how developing smart surveillance tools and across the region, and fosters cross-border collaboration and data sharing, leverage on cutting-edge technologies in breeding and MLN resistances, enhance farmers' education and local capacity building, focus on sustainable management practices, and together we can safeguard the maize production, food security in sub-Saharan Africa. Our goal is completely ensure that MLN is free from Africa and serve as a global model for managing plant health threats. I sincerely thank and congratulate all the partners for supporting this entire mission in Kenya and Tanzania, Rwanda, Uganda, Ethiopia, Malawi, Zambia, Zimbabwe, and Bill and Gates Foundation, Minnesota, and then several other partners. And with this, I sincerely thank the organizers for this. Yeah.
Thank you very much, sir. Yeah, thank you.
Open for any suggestion or questions or interactions.
Any question will be mailed to you. Thank you, sir. You can leave. Thank you, sir. Now we invite Dr. Asana Oruj. Dr. Asana Oruj, Senior Professor and Chairperson, PG Department of Food Science and Nutrition, University of Mysore. She will be speaking on translational research exploring anti-diabetic potential of Morus indica. In the meantime, going to have some.
Right. Okay. Ladies and gentlemen, we have one important announcement to be made before ma'am starts. So we have thematic lead and invited talks as well as oral presentations at 3 different venues. And I request the students who are participating or the chairpersons or co-chairs to please leave for their respective venues. The theme A is the frontiers of technology. Venue is Moot Court at the 5th floor of the same building. And there we have presentations on digital plant pathology, AI, ML, and smart surveillance. The second topic is the omics revolution, genomics, transgenics, and gene editing. The third is nanotechnology and next-gen therapeutics. I request the participants to please leave for the venue as we will be starting parallelly with these sessions. The second theme is integrated disease management and ecology. The venue is multipurpose hall that is at the basement of the same building. So the topics that will be covered is intensive IDM, IPM, biocontrol, endophytes, and green chemistry. The second one is soil health, nematology, and root microbiome. The third one is ecopathology, natural farming, agroscaping, and climate resilience. The third theme is food security and value addition, which will be conducted in the conference hall that is on the first floor of the same building. The topics covered are post-harvest pathology, mycotoxins, and food safety. The second one is applied mycology, innovations in mushroom cultivation and nutraceuticals. I request the participants as well as the chair and co-chair of the sessions to please leave for their respective venues. However, the talk or the plenary lecture of Madam Uruj will be continued. Thank you. Thank you.
A very good morning to everyone. Thank you, Chair and Co-Chair. At the outset, I would like to place my gratitude and heartfelt thanks to the organizers of this conference, and in particular to Dr. Shalini Singh for including me in this event. I come from the erstwhile Royal University of Mysore. It is a 110-year-old university. You would be wondering what a person from nutrition background is doing in a plant pathology and mycology setting. My only connect between your organizers, the theme of this conference and myself is that my interest in medicinal plants. I've been exploring the therapeutic potential of several medicinal plants, local and also from the Western Ghats region. So in the time allotted to me, I will be exploring, I will try to share what was our finding with respect to the anti-diabetic potential of Morus indica, the mulberry leaves. This is a glimpse of my university. I don't have time to go through. Anyways, as we are all aware, plants are playing and have played a large role in treating human health. Many drugs have been manufactured from medicinal plants. So with that background, I'm talking about my research. And for this, I just want to spend a minute and tell about, we need to adopt the translational research methods like in vitro, ex vivo if possible, and then animal studies, which comprise preclinical, and then move on to clinical studies to actually, project and understand the real therapeutic potential of medicinal plants. So one of the major aim of my research from my master's days has been diabetes, exploring different aspects, management, dietary management, nutritional management, and then developing functional foods. And then later I have strayed into medicinal plants. And this has been the major area of my work for more than 20 years. So we have tried to identify and validate local traditional plants used in the management and treatment of diabetes. So my team has explored more than 24 different types of medicinal plants. And among that, I'm going to share only on Morus indica. We have adopted the preclinical to clinical, that is T1, T2, and T3 model to understand and explore the anti-diabetic potential of Morus indica. So in preclinical, we have to adopt the in vitro, both at the enteric and the systemic level. Then followed up by you can use some ex vivo methods, like you can use the yeast cells and the hepatocytes, and you can use the hemidiaphragm of the rats also to understand the glucose uptake potential. And finally, as part of preclinical, we can conduct the toxicity studies to establish the safety, and then move on to the adoption of the STZ, streptozotocin-induced diabetic model rats. to understand the anti-hyperglycemic potential of the plant extracts. So here, as I said, I'll be focusing on Morus indica. We all know that mulberry leaves are very popularly used in silk production. They are fed to the silkworms. Apart from that, there are some therapeutic uses of Morus indica. And in preclinical, what we have done is, first is we established the antioxidant potential using different model systems, both in food and biological systems. Then we use the in vitro model systems to establish whether it has got any glucose adsorption capacity and can it retard the glucose diffusion in a dialysis model. And we found that it has higher glucose adsorption capacity, and it can also retard the diffusion of glucose across dialysis membrane. Then we also used the yeast cell membrane and isolated the rat hemidiaphragm. In that, we could establish that it has enhanced glucose uptake, and this indicates that it may enhance the peripheral utilization of glucose in the target cells. We also explored the development of some functional foods by incorporating the Morus indica powder. And ex vivo studies, we were surprised and very happy to note that it has the ability to inhibit all the enteric enzymes, especially alpha amylase, glucosidase, beta glucosidase, and sucrase in a dose-dependent manner. Further, we have used certain techniques to isolate and extract the bioactive compounds, and we could identify in 80% methanol extract, though it was present in aqueous and other extracts, we found that chlorogenic acid, ferulic acid, rutin, and epigenin, one of the important bioactive, were present in these extracts. With this, we moved on to undertake the animal studies. First one was we tried to establish how safe it is. We did the toxicity studies as per the OECD guidelines. After 14-day treatment at the maximum dose of 2 gram per kg body weight, we found that there was no mortality or toxic reactions, no changes in physical or behavioral changes in the rats compared to the control group, and there were no deleterious effect on metabolism. As indicated by the oxidative stress indicators, liver enzymes, lipid profile, and hematology. So we could establish that it was non-toxic in nature. And we undertook a pilot study for 2 weeks, and we fed the rats which were induced with diabetes by injecting streptozotocin, and we found a 38% reduction in the fasting blood glucose levels after 15 days. So armed with this, we further conducted a long-term anti-hyperglycemic study in 5 groups of rat, comparing with the standard drugs like insulin and glibenclamide. And in that also, we found that the Morus indica extract was as effective in reducing the postprandial glucose, and also at the end of the 8 weeks, we found that there was a reduction in the blood, not only blood glucose, but also in the lipid profile also. So anti-hyperglycemic studies established that it has got the potential to reduce the blood glucose levels in diabetes-induced rats. And we also conducted anti-cataract, that is anti-glycation studies in another study. After 6th week, one set of rats were studied for their histopathology in liver, kidney, and pancreas. Then the next set of rats were continued for another 2 weeks, and then later we conducted the anti-glycation studies, and there we found that there was a delay in the cataract formation compared to the untreated rat group, wherein there was already cataract formation because of the uncontrolled diabetes. So armed with this, we moved on to conduct a clinical study in type 2 diabetes subjects. This was a randomized, single-blind, free living study with no changes in medications or lifestyle pattern. involving a diabetologist, and this was conducted for 8 weeks. The maximum dose as established from the preclinical studies given was 6 grams per day in 2 divided doses. We took permission from the Human Ethics Committee and then proceeded with this. We had 4 groups of subjects. From among 80 type 2 diabetic subjects who were screened, we finally narrowed down to these number who fitted the inclusion criteria. One group was subjects on oral hypoglycemics. They were provided with placebo. And the second group, they were on oral hypoglycemics. They were provided with Morris tablets. And then there was a third group who were on insulin. They were provided with placebo. And the fourth group treated on insulin and supplemented with the test material. So after 8 weeks, we were happy and surprised to note that there was a significant reduction in fasting and postprandial blood glucose, and also the long-term indicator glycosylated HB also reduced, and there was a good anti-hyperglycemic effect in type 2 diabetic subjects by augmenting serum insulin. We analyzed even the serum insulin levels at the end of 8 weeks, and we found that it was as good as or comparable with the control group, which was treated with insulin. And another thing is that we were surprised to see that there was a significant reduction in the serum total cholesterol and triglyceride in both the experimental groups. The subjects were happy with these tablets, and they did not develop any kind of gastrointestinal discomfort. It was well tolerated, as indicated by the renal and the liver parameters. The urea and creatinine and even the liver enzymes remained unchanged in both the groups. So this was one of the single blind study. That is, the subjects did not know whether they received the placebo or the plant material. So this single blind study in type 2 diabetes subjects validated the observed anti-diabetic properties of Morus indica, as we saw in the preclinical studies, as indicated by the preclinical studies. So I would like to say that by adopting this translational approach, we were able to identify some of the anti-hyperglycemic attributes. That is, the plant has the ability to lower blood glucose. It increases the viscosity. That is why there was a glucose diffusion inhibition. It modulates the enzymes of carbohydrate metabolism, promotes glucose uptake, and in the animal and the human studies, we could see that there was an increase in the insulin secretion and synthesis, and it is a well-known antioxidant. So some of the bioactives we could identify was apigenin, which is responsible for this. Besides that, the plant, that is the leaves of this plant, they are good source of viscous dietary fiber, and the polyphenols present perhaps are responsible for increasing the viscosity and preventing the transport of glucose across the intestinal membrane. So we propose this mechanism of anti-diabetic action, as I said, by increasing the viscosity of the intestinal contents. So adsorption of glucose molecules, and as indicated in the in vitro methods. And then second was potent inhibitor of all the enteric enzymes, and thereby it delays the release and absorption of glucose into the bloodstream. Third mechanism is what we could identify from the animal studies, is that it modulates the major carbohydrate metabolizing enzymes, that is both the glycolytic and gluconeogenic enzymes, that is G6PD, hexokinase, and it inhibited aldolase and glucose 6 phosphatase, and this blunted the glucose rise. And the fourth mechanism, as we could identify from the ex vivo studies, is that it improves the glucose uptake and transport across target cells, supporting that it will promote the peripheral utilization of glucose. And lastly, there is regeneration of pancreatic beta cells, as indicated by histopathology studies, and also supported by the observation that there was an increase in the insulin levels in the treated rats and also in the human subjects. So these are the various mechanisms of action and other aspects also we have explored. I have not included that. We have also conducted anti-hyperlipidemic studies both in animals and in human subjects, and anti-cancer studies and anti-glycation. So we are yet to continue with this. Some of my students are still working. I have superannuated, but I still have some of my students continuing with their PhD work. And I'm very happy to say that those who are part of this work, they're all well established, but they're still continuing with this work in their respective institutions. So what I would say is many of us are interested in plants, right? Apart from plants serving as food for us, plants serve as, they have medicinal properties. We know that we have been using in Ayurveda, in Unani, in everything, there are plants used. But we have to explore our plant kingdom. What is most important is, as a scientist, it is we need to know the mechanism of action rather than just suggesting to subjects, to people, that just consume it, because without knowing how safe it is, it might end up in some disasters for the subjects. Diabetics are very desperate to try anything in order to control their diabetes. So we need to adopt sound scientific methods in order to establish the medicinal properties of medicinal plants. Thank you very much for this opportunity. I thank my arteries, who are, as I said, are very well established in various institutions, not only in India, abroad also. Thank you very much for this opportunity.
So, thanks, Mana, for this very good lecture. And as we know, plant can grow without human intervention, but human cannot live without plants. And if there is plant, there is disease. That disease is a rule and health is an exception. And here in SMPP, we work on understanding the diseases, their interaction, the environment, and other. And this platform gives us opportunity to authenticate our work and research and other things, and make a platform to present our data and our knowledge and make documented for further generation. And I request my co-chair and co-fellow to give their comment.
Thank you, all the participants. All the presentations were very good. They were insightful, and I hope all the participants will take cues from them. Now there will be we will wait for the tea. The facilitation will be carried out later on by, as the Pro Vice Chancellor has desired to facilitate the participants, facilitate the speakers later on. We will break for the tea. Thank you.
Thank you, sir. I thank Dr. Manjul Pratap Singh, sir, Dr. Ayako, and Dr. Alpana Srivastava for taking up the entire tenure of the session. And I also thank Dr. Pratibha Singh, Dr. Shiv Kumar Agarwal, Dr. L.M. Suresh, Dr. M.D. Arif, and Dr. Asna Uruj ma'am for their enlightening lectures. May I now request the attendees to please move to the thematic sessions which are taking place at 3 different venues. I repeat, one is in Conference Hall, first floor. The other one is in Moot Court, 5th floor. And the third session is in basement, that is multi-purpose hall. So I request all of you to please move to the venues for the thematic lectures. Thank you so much. We will come back for the post-lunch session at the auditorium venue. Thank you, everyone. And our Pro Vice Chancellor, sir, will be joining us. whenever, he is free, and he'll join us to take up the session for felicitating our guests. Thank you.
You want to speak?
No, no. I welcome all the eminent plant pathologists to this auditorium. And I really appreciate the efforts of Professor Shalini Madam and all other members who are associated with ISMPP. I also welcome the Pro Vice Chancellor, Anil Sir, and many other distinguished scientists who are here. Now the first lecture is by myself only, so I would like to go ahead with that part.
Yes.
हो गए ना? तैयार हो गए? I wanted to concentrate. पहले registration करना चाहिए। या ना? Speakers को वो साफ जा रहे हैं ना अनिल साहब आप शुरू करूं प्रेजेंटेशन बाद में हाँ और हिल स्टेशन करते बोल रहे हैं मैडम प्लीज कम वो हिल स्टेशन करना चाह रहे हो ना
हाँ जी सर वो करना चाह रहे हैं
अनिल साहब को बुला ले फिर ऊपर
सर अभी बुलाओ हाँ जी बुलाते हैं सर
hill station to the speakers. The organizers would like to felicitate all the speakers first and then proceed further. So please bear with me for some time. On behalf of ISMPP and Amity University, I welcome the dignitaries, the Pro Vice Chancellor sir and all other professors and Professor H.B. Singh, the advisor to the government of UP and many others. Oh, it's
ठीक है सर, आप अनाउंस कर दीजिए, आइए सर। हाँ जी, आ रही मैडम, आ रही। हाँ जी, ओके। To give away the Lead Lectures Award, may I now invite our Honorable Pro Vice Chancellor, Sir, Dr. Anil Vashisht on stage. I request Dr. Shalini ma'am and Dr. Manohar Acharya sir to please accompany.
It was a pleasure yesterday and it's a pleasure today meeting you.
Okay, I invite Dr. Shiv Kumar Agarwal sir to please receive his award.
Sir, it's a pleasure.
Dr. Shiv Kumar Agarwal sir. Give him a huge round of applause, please. I request the audience to please give a huge round of applause as we are felicitating him. So we are honored to have you all here. May I now request Dr. L.M. Suresh sir. Oh, okay. Sir ne naam likha, so I thought. Now I request Dr. Pratibha Singh ma'am to please receive the award. Dr. Pratibha Singh ma'am. Felicitation. Ma'am, your presence is very precious for us. Thank you, ma'am. I request Dr. Manjul Pratap Singh, sir, to please accept our felicitation. Dr. Manjul Pratap Singh, sir. I request Professor Dr. Ali Anwar, sir, to please accept our felicitations. He's not here. May I now invite Dr. Yaqub, sir, to please accept the felicitation. We take pride in presence of such elite scientists at our campus. I now request Ma'am and Sir to felicitate Professor Dr. Chakravartula Manoacharya, sir. Yeah. Sir is a NASI senior scientist and Platinum Jubilee Fellow. I request all to please give him honor with your claps. Now I request Dr. J.C. Rana, sir, to please accept the felicitations. May I now request Dr. Muhammad Akram, sir, to please be on stage and accept the felicitations. I now request Dr. Daisy Basant Rai, ma'am, I now request Dr. Debasish Chakrabarti, sir, to please accept the felicitation. And it is giving me immense pleasure that I am calling the names of such eminent scientists Now I request Dr. V. Celia Chalam, ma'am, to please come up on stage. I request Dr. Deepak Singh, sir, to please accept the felicitations. Yes, sir. Now I request Professor Dr. Rakesh Pandey, sir, who is an eminent scientist at CMAP, to take the award for PR Verma Award 2025. I invite Professor Dr. Rakesh Pandey, sir. अवार्ड यहाँ पे नहीं है इन्होंने कहा था लीड लेक्चर्स में ही है कंटिन्यूएशन नहीं सीनियर मैम को दिया ना उन्होंने कहा जमीन की अच्छा 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 क्योंकि सर है ना सर मिल जाए बस हम सारे हो गए सबको सबको मैम का दे दीजिए तो For PR Verma Award, I request Professor Dr. Rakesh Pandey, sir, to please accept the award. It's an honor for the Amity family that we are conferring this award. Now I request all the awardees to please come up on stage for a group photograph. I request all the awardees for a group photograph. I request you to please be on stage as we have the photographer right now with us. So I request all to please be on stage. Thank you, sir. मैम आप लोग आ गए प्लीज आप चाहिए ना आप तो सूत्रधार है नहीं Thank you, everyone. Thank you for making this occasion special. Thank you, ma'am. अरे नहीं सर खा लिया सर नहीं सर थैंक यू सर Ladies and gentlemen, we'll be continuing with our lead lecture session, and I now request Professor Dr. Manohar Charya sir to please continue. Sir. Stage is all yours.
On behalf of all of us and on behalf of Usman University Hyderabad and on my own behalf, I would like to felicitate Professor Anil Vasist sir, Vice Chancellor. I request him to come onto the dais.
I was waiting, ma'am, coming.
This is from society, I think.
Ma'am, I see.
And on behalf of all of you and on my own behalf, I would like to felicitate Professor Shalini Singh for all our efforts and making this particular program a grand success, and I wish you all the best. Thank you so much, sir.
Congratulations, madam.
Thank you, sir. Thank you so much, sir.
Good afternoon, all of you. Now I welcome Professor C. Manohar for his lecture. Professor sir is going to talk on sustainable mushroom production using agro waste employing circular bioeconomy. Sir, over to you, sir.
You can start from.
Now, the topic that I chosen is sustainable mushroom production using agro waste, a circular bioeconomy model. And actually, Professor Shalini Singh, madam, and the university, Amity University authorities, and ISMPP, and Dr. H.B. Singh, I would like to thank all of them for having given me an opportunity to be with you. And this particular topic that I have selected because of the fact that Professor Shalini Singh has been working for the last so many years in this particular field, and she has been doing a lot of service to the society in this regard. And also Amity University has got a very good name and fame in food product, in the food and as well as sustainability of various activities, and therefore food and agriculture sustainability. So I have selected this particular topic. Now India being highly populated, and it has been suffering with hunger and so on and so forth, and greater problem being the food supply to the people, and malnutrition is very high, and many people are suffering with that kind of malnutrition, maybe around 600 million people in Asia. Therefore, keeping all these things in view, we have to look for the various avenues, and that comes into existence and that are there in the scientific field. And this is one of the aspects that I have taken up is the organic waste. And in view of the energy store shortage, it needs to be conserved through efficient management of waste, agro waste especially, or the other resources. Now, recycling of waste and its efficient utilization is very important. It will help to conserve not only the national resources, but also useful products like fertilizer, fuel, and feed, and other economically viable products can be made available through this particular organic waste recycling. So it also combats the pollution and all the, you know that in recent, the pollution in Delhi and other activities that you could see that there is a lot of problem because of air pollution and burning of the crop residue. So the possible alternatives to improve nutrient status of organic waste, vermicomposting, and mushroom cultivation is the promising scope to increase agricultural bioproductivity and microbial food production. Now we have, these are the types of organic waste that we have got. Crop residues, that is straw, stalk of various crop plants. And in the case of pulses, it is husk and shells, and oilseed shells and husk and others. And in the sugarcane, it is the baggage, and the cane and crash, and also fruit fields, coffee hash, rice straw, and all other things, horticulture waste, and so on and so forth. And in the case of animal waste, you have got dung and poultry litter, and so many other things, aquaculture waste also. And we have been a lot of in use, that is fertilizer bags, pesticide containers, plastic waste, and this has become a major problem of recycling the plastic and storage materials. So all this has to be utilized for the recycling, and as well as we should see that how best one can see. So at a global level, it is around 680 million tons per year, the agro waste that accumulates, and 300 to 380 million tons of organic waste is released at global level. And India is number one in producing a lot of agro waste that is released for a year, around 700 million tons. And the global wastage that is generated is per year in the whole world is 3 to 5 billion tons. So India being the one of the biggest agro waste producer, it is necessary that we should combat the pollution and we should utilize this particular agro waste as a biofertilizer or by recycling it to various other products. So the action results in air pollution, greenhouse emission, loss of biomass and resources if it is burnt away or if it is underutilized or unutilized. So these are the crop residues. And of course, throwing the organic waste or litter, etc., in somebody's house, some people will take the advantage and they will be burdening the pollution in others' houses also. And this kind of unethical issues, I think one has to look into this, and we should be very careful with reference to our not only global waste, but India is being rich in this producing a lot of waste materials. We have to look for this. Otherwise, I think we'll be suffering a lot, not only from the diseases, but also in the next few years, we'll be suffering with the climate change and as well as the pollution and so forth and other things. So why mushroom cultivation? This is the excellent source of good quality proteins to fight protein malnutrition in the Indian masses. And it is the highest producer per unit area, and its time is also less. And it is profitable, especially to the women folk. And as a rural industry, it got established in India. And environmentally sustainable way of recycling this agro waste. And it helps to reduce pressure on arable land because it can be grown in indoors, utilizing vertical space. And excellent medicinal value it has got, useful for diabetes, cardiac diseases, anti-cancer, and labor intensive. And all of you know that we are not able to provide jobs and other things. And it is necessary for us to request or make the youth to go for this particular mushroom cultivation, and so that they can get gainful employment. So foreign exchange earner through exports. India is around 0.35 million tons is produced every year. This is the mushroom production, and in the whole world, it is around 60 to 80 million tons that is produced, and that is how India, and when you compare India with other countries. So lot of qualities are there with the mushroom cultivation, and these are some of the edible mushrooms. The most important one is the button mushroom and the Lentinus edodis and Valvariella padistra mushroom, and then so padistra oyster mushroom and Pleurotus flagellatus, ear fungus, Auricularia auricula, and also Calocybe India milky white mushroom, and especially button mushroom, padistra mushroom, oyster mushroom, and milky white mushroom have become very popular in India and elsewhere, and a lot of money. There are millions of tons of these edible mushrooms are sold in outside India, and of course it is picking up in India. In 2015, it was only 0.16 million tons, whereas now it has reached 0.35. So termite mushroom, Termitomyces, of course, the difficulty with this is not grown artificially. Still difficulties are there in cultivating this particular mushroom, but it is a very good mushroom from the point of edibility and other qualities. And Marcella esculenta marl, under apple orchards it will be growing, and
From Italy. So that also we will have to add in our session.
you will add.
So we could.
Yeah, so Marcella esculenta is under apple orchard. This marl is also very difficult, but India is getting 4,000 crores by exporting this particular marl, and this is one of the best edible fungus that is getting through which money is earned for India. Tuber melanosporum, highly useful, not only edibility, but also from medicinal point of view. So Lentinella edodis and lengitis, 50 grams of lengitis is sold in northeastern India for 200 rupees. And that is the quality of this particular edible mushrooms. And now highly marketable products are coming out of this particular one. And Flammularia enoki mushroom. So these are some of the mushrooms: button mushroom, Valvariella valvaceae, and then Pleurotus, Calocybe indica, the milky mushroom, Termitomyces, and Marcella. Now these are the medicinal mushrooms. Ganoderma, it boosts immunity and anti-cancer, and there are also Cordyceps, it is energy tonic, and Enotus, antioxidant, and Tramatus is immune booster. And like that, we have a number of Griffiola. We have the right side photograph that is very good medicine for heart diseases and other such kind of things. And we have poisonous mushrooms, and like Amanita phalloides, it is a death cap, and Amanita muscaria, and all these things are poisonous mushrooms, but their metabolites, alkaloids, are very highly useful in the form of medicine, and these are the alkaloids can be extracted, and metabolites which are secreted can be made into medicines. I don't want to go into the other things, and it takes a lot of time for me. So this is the mushrooms nutritional value. You can see, read them just like that: energy, protein, carbohydrates, fiber, fat, vitamins, and all that. This is a very good healthy food that is available. Now this is the profile that we have got. Now these are beta glucans, immunity, antioxidants, polysaccharides, which are also, some of them are anti-cancer and low caloric food, cholesterol free, and it improves gut health also. These are some of the aspects, and because of that, the edible mushrooms have become very important in the dietary and all that, so on and so forth. So the step by step, all of you know this particular one. First, you should select the mushroom and then substrate and then spawn pasteurization and then spawning. Good spawn, healthy spawn is necessary, and filling and incubation, all these things are important. And then casing and fruiting, harvesting, every point one has to be very careful at every moment. And of course, around edible mushroom will fetch you around 20 lakhs per year, and all other mushrooms will be fetching the housewife or in the industry around 8 to 10 lakhs. So this has become a rural industry now in India. And then these are the spawn, and you can see the production of all these mushrooms. Now, bioconversion is the transformation of organic waste into valuable products, and bioconversion of agri-waste into useful products, bioplastic production, pharmaceutical compounds, and it is also converted into animal feed. It is also natural organic waste. After the spent mushroom will be a very, very good useful as a fertilizer. It is a natural decomposer, and they decompose organic waste, and mushrooms are also known to contribute a lot for this, and waste is reused in this particular one. So why using mushroom for waste recycling? They decompose agricultural and organic waste, and they convert to low value agri waste into nutritious edible biomass, and reduces environmental pollution, supports circular bioeconomy, and spent mushroom is that is substrate is used as organic manure, animal feed, and warming camp. It is a low-cost technology, eco-friendly, and sustainable technology. Now, this is a sustainable concept integrating biological resources into closed loop system, and it emphasizes more on resource efficiency, reuse and recycling, and as well as the reducing the reliance on external inputs. It is a nutrient loop closure, and waste is transformed into valuable resources. So this is one of the important aspects in the case of bioeconomy, and one can earn a lot in this particular one. So spent mushroom is organic manure, and it is also used because it has got nitrogen, phosphorus, potassium, and also it improves soil fertility, enhances soil structure, water holding capacity, aeration, root growth, nutrient cycling, and it also suppresses many diseases, and it contributes to sustainable agriculture, and it's a low-cost alternative to chemical fertilizers. So applications of spent mushroom is animal feed, it is soil condition and biofuel source, bioremediation. It is also cultivation after microbial processing, still you can use it, and mulching also is one of the important aspects. So microbial inoculum, and it is a starter for many of the activities. Industrial enzymes can be produced from this particular one. Bricks and boards and insulation materials can be prepared out of this, and biochar also prepared, and potting media is also used. So like this. So this is the last slide, I think, and the relevance to India is the practical application of the circular bioeconomy, where agriculture residues are high valued food, that is mushrooms, and valuable byproducts are, this is the spent substrate. It is efficient waste management, it is beneficial, income generation is possible, nutritional security is possible, employment opportunities are created, resource cycling is possible, climate resilience is because of the spent mushroom as organic waste recycling is possible, scalability and adaptability. So I thank every one of you for having given me this tiny fungus an opportunity. Thank you very much. I thank the organizers, and no country can progress without agriculture and education. And knowledge is important. Knowledge is power, and until and unless you do this particular one, and if you don't go for things like this, I think it is very difficult for us in India to feed the growing population in the years to come. Lastly, I would like to say the razor blade is very sharp, but it cannot cut the tree. The axe is very strong, but it cannot cut the hair. So, everything is important according to their unique purpose and properties. Don't look down anyone, and don't look down science, and don't look down mushrooms. Thank you very much for having given me an opportunity.
Professor sir has spoken very good talk on wealth from waste and also circular economy of the country. Now topic is open for discussion. Any questions, clarifications? Well, if there are no questions, on behalf of all of you and on behalf of the organizing committee, I thank Professor Chakravartila Manorachari for having delivered a very good talk. Thank you, sir.
Now you have online? Koi hai online? And the second one? Madam, should I take second one?
Yes, sir.
That is which one? Rana. But she is there. It is online or what? Okay. See, Dr. J.C. Rana, Country Director, the Alliance Biodiversity International and SIAT, she is going to speak on natural farming as a strategy for, Dr. Rana is going to speak on natural farming as a strategy for ecopathology and sustainable plant disease management. 15 minutes, sir. Thank you.
So, good afternoon to all dignitaries on the dais and off the dais. I am not a plant pathologist basically. I am a plant breeder and working on more on agrobiodiversity for the last 35 years around. I'll be talking something on natural farming in ecopathology and plant disease control, that how it can help us. Though it's a new science as of now, I would say, natural farming is being talked about a lot, but if you go back to our own graduation programs and all this, so we have never been taught on natural farming. This is quite a new concept and a lot needs to be done. Sorry. Thank you. So, as you know, that natural farming, it's a kind of chemical-free farming, what we talked about nowadays, and ecology-based approach that built on certain principles of natural cycling, what was taught in the earlier lecture, and how we can use local and own farm resources. That's why sometimes it is called zero-budget natural farming, and how harness the beneficial microbes and biodiversity, which increases soil microbes and mixed cropping, agrobiodiversity to enhance nutrient cycling. Soil health is one of the important component which is related to agroecology or natural farming, and overall enhancing the plant resilience of the system. So why we are looking for natural farming and ecology balance in agriculture, that the kind of depletion of resources that has occurred at this stage. So we need self-regulated system where we can take care of the soil, plant, microbe, insect, all the kind of interaction that they have, and natural suppression of pest and pathogen predators and the competition. It reduces the ecological disturbance and also enhance system resilience and stability. For example, predatory insect controlling aphid in diversified cropping system, soil microbe competition, reducing fusarium, rhizoctonian, and many incidences. So natural farming for disease management, how it helped that. So it is a preventive approach, not a chemical curative approach, that how we are going to prevent. And in today's context, whether it's a human beings or a plants or animals, we are talking more on a preventive measure rather than a control measure. And how we promote the beneficial microbes using inputs like a lot of inputs, Jivamrit, Bizamrit, a lot of have been done. Neem extract and crop diversification, as again been talked about, that intercropping, mixed cropping, and all these kind of cropping system that have been talked about for natural farming, and mulching, etc., which is not a new thing. So, for example, we have a neem-based formulation now that suppress fungal pathogens, Jivamrit enhance microbial activities, and intercropping enhance this lower the disease spread. And we know that role of indigenous microorganism, that how promotion of native microbial communities, natural biological agents, and the competition for resources, because indigenous microbes compete more with the pathogen for nutrient and space, so limiting their establishment and growth. And how we are production of protective compounds, induction of plant disease mechanism that trigger new systemic resistance, enhancing the plant's internal defense system, and site-specific competition effectiveness. So common natural inputs for disease management, you already know that Jivamrit, biological extract, cow dung-based formulation, that fermented extracts, and the key benefits that give you in the natural farming, that how this plant pathogen naturally improve plant health, vigor, enhance soil biologic activity, and eco-friendly and cost-effective. So when you compare these natural inputs and chemical pesticide, then there are a number of benefits that I've been talking about in the last, but in the summary, that how their mode of action, impact on soil health, waste and development, environmental impact, sustainability, plant health, and how residual and the food and farmer dependency, this how it's the overall benefit over the chemical pesticide. Coming to ecopathology, and when we say ecopathology, that how natural farming helps in disease management, so it's a kind of holistic plant disease management that takes care of plant health within the entire ecosystem, not just isolated host pathogen interaction, which is happening sometimes when we look at this segment in silos. Sometimes where a plant pathology, so you look just only plant pathogens and entomology, then plant. So something that is happening, so which is not required now. People are talking about the entire holistic approach. We bring all together and look at the system approach, not really on in silos on a compartmentalized wise. So multi-factor interactions are focus on dynamic relationship among plants, pathogen, microbes, climate, and farming practices, disease as an ecological imbalance, prevention over cure, as I said, reduce chemical dependency, and foundation for sustainable agriculture. Sorry. So this is the ecological basis of disease suppression, that healthy soil is always, if there's a healthy soil, it is the first defense against any disease and pest management. Microbial competition and antagonism, balanced nutrient cycle, induced plant immunity, agrobiodiversity as a barrier, and improved soil structure and aeration. We have done some case studies through our partner in the Himachal, and there are some case studies which have been done how it has helped in the controlling of the thrips in the chili field, and there was a problem with the chemical farming. Then they go for a natural farming interventions, and some of the results which are produced, that they are quite encouraging result, I would say, not excellent, but encouraging, because they are small scale experiment, and natural farming have been tested at this stage only under a small experimental stage, not a large farm. Similarly, for soil-borne diseases, how we can, by Jivamrit and all this has been studied, and a small case study have been created. For such gaps and future directions are there, that there are a number of gaps which have been identified in the natural farming or this, because there are a lot of lack of evidences, there are a lot of lack of capacity building, and many things which are revolving around when we talk natural farming in a holistic approach. For this, probably I just want to touch few slides, three slides, that we are developing a kind of Himalayan agroecology initiative. It is more on a Himalayan agroecology. We are 3 countries, India, Nepal, and Bhutan are being focused, and for Indian Himalayan region, we with the UNDP and some national partner, we are developing a kind of roadmap that by 2047, how it looks like. Because when we say transparent food system, that is must. It's not an option now. We cannot play with the environment and climate and the soil anymore, or the water anymore. So we need a kind of food system that has to be transformed now from the present to a kind of sustainable food system, because they are holistic, resilient, and rooted in ecological balances, all which I talked about in my few slides. So it's summarized here that how agroecology food systems are going to serve. The agroecology, how it contributes to sharpen the Viksit Bharat idea, that it aligns deeply with the transformative goals of the Viksit Bharat, that the entire roadmap, we have developed 3 phases, phase 1, phase 2, phase 3, accordingly until 2047, that how we achieve some of the goals related to this. So this is the architecture of this roadmap, that visioning and need for action, rationale, strategic pillar, objective, and then implementation plan is there. So these are the 7 pillars which we have identified, that if you work on agroecological ecosystem, that then you need to work on these 7 pillars. So first pillars come from the governance system, that how government policies are going to impact on this. Then how you are ensuring the enhancing access to land, water, and healthy soils. That is another thing. And then how you ensure the climate resilient and agriculture for healthy ecosystem. Then 4th is how you promote sustainable, sufficient, healthy, and sustainable diets for all. Then build a fair, shorter, and cleaner supply chains. Then how we reduce this food loss and waste. And 7th pillar is putting trade into the service of sustainable development. So these are the 7 pillars, and all the pillars have been defined, that how food governance, what we need, ensuring land, water, soil, what we need, climate resilience, and for healthy diets. So similarly, these are the 5th, 6th, and 7th pillar. And the cross-cutting aim is that how we translate implicit sustainability objective within the existing policies into actionable agroecological results, addresses policy inconsistencies. Because what we found, we did a lot of, I did more than 100 policy analysis we did through 6 consultants, and we found that there is a lot of inconsistency in the policies. Policy overlaps are there, policy gaps are there. So how we are going to address those? So this is that how to improve existing investment on aligned to this SDGs. And to conclude, that natural farming is really required for restoration of soil and biological health, strengthening plant immunity, and enhances agro-ecosystem biodiversity. And the key away would be that shift from, as I said, the control to prevention, and system-based sustainability is required if you really want to achieve all those things what we're talking about today is in the natural farming concept or organic farming. Thank you very much.
Thank you very much, Dr. Rana sir. Rana sir has given very exhaustive information on botanicals. Neem plant itself is known to have 200 chemicals. And biosecurity of agriculture products in India, in developing countries, is very important. And in this regard, I request some of you to, if you want to question him or ask some of the suggestions from him, you can. Anybody, please? If there are no questions, I thank you very much, Dr. Rana sir, for having given exhaustive account. Now I'm coming to Dr. Mary Lucy Arange, Senior Scientist, SPS, Nairobi, Kenya. She is going to speak on strengthening biosecurity and trade by improving pest risk prioritization and response mechanism in Asia. Dr. Chalam, madam, you are there. Okay. Sir, please. Online. This is online. Dr. Mary, madam, you are welcome, and you can start your presentation, and 15 minutes is given to you, ma'am.
Thank you very much, the organizers of this particular conference on pathogen, plant health, and food security, allowing the various participants to share their recent advances for climate and resilient agriculture and landscape conservation. My name is Mary Lucy Oronje. I am Senior Scientist, Sanitary and Phytosanitary at the Africa Office in Nairobi. In this particular presentation, my colleague has been instrumental in the preparation, and this is Dr. Manju Thakur. Today, we are going to share with you CABI's interventions in strengthening biosecurity and trade by improving pest risk prioritization and response mechanisms in Asia. Let's hear about CABI, because many of you may not know about the Center for Agriculture and Biosciences International. We are an intergovernmental international organization that has been set up by the United Nations Treaty, and we operate among our 48 member countries. CABI is a global leader in scientific publishing. That is the CABI abstract, the journals, and some of you have interacted with this. Also a key leader in agricultural research, especially biological control, new pest, and general pest management. We are also involved in agricultural development, and this is multi-country projects, and also from production to international trade. We are also involved in biosciences. This is diagnosis, alternative methods for pest control, especially in the use of biological control agents and low-risk pesticide alternatives. We also do project management from design to implementation of various projects that CABI has been involved in. In Asia, I think maybe it's important to mention that in Asia, we have our member countries is Bangladesh, Brunei, China, Malaysia, Myanmar, Pakistan, Philippines, Sri Lanka, and Vietnam. In any case, if your country is not a member country, current member country of CABI, please, you can register your interest, and then we can make follow-up. The topic that we wanted to share with you concerns biosecurity, and this is defined by FAO as strategic and integrated approach that encompasses the policy and regulatory frameworks, including instruments and activities for analyzing and managing risks to human, animal, plant life and health, and also associated risks to environment. Specific to plant health, we can therefore define plant health biosecurity as protecting plants from harmful pests. When we say pests, look at pests as anything injurious to plant life. And it is instituting measures that prevent these risks from invading our plant health system. We're looking at biosecurity. There have been biosecurity concerns in food safety, in animal health, but critical in this particular discussion, we are going to focus on plant health, so plant health pests. Of recent, you are all aware about the invasive species that have continually invaded the region, Asia, Africa, all the continents. A number of them have, in the recent past, we have seen plant health, invasive weeds, for instance, continually invading our ecosystems and therefore disrupting plant life. Also, we have invasions of insect pests. And recently, I think we know about the fall armyworm that did invade a number of continents across the globe, including Asia. And this invasion by insect species are increasing exponentially. Same to also the invasive weeds. But apparently invasion by mammals, we are talking of, the black rats and others, have been decreasing. And the reasons why this has been happening is because of, sometimes through our travels, through our trade, especially when it is not safe, we are key facilitators of some of these insect pests, some of these weed invasives into other areas. The borders are porous. There's weak biosecurity to be able to even identify these risks in advance and even to stop their spread when they are identified. There's lack of capacity to even eradicate some of these pests, and therefore we end up having new pests in new regions devastating the agricultural systems across countries in Africa and across countries in Asia and the globe. This particular invasion end up affecting our production, end up affecting our trade, and overall the economies of the countries that they invade. But I want to bring your attention to what we call the generalized pest invasion curve. This is important to be able to showcase the importance of pest prevention as opposed to pest management. And when I say pest management, kindly notice the difference between pest risk management and pest management. In this case, in this invasion curve, I'm talking about pest risk management, that is prevention, eradication when they occur, versus pest management, what we call in this particular curve, the asset-based protection. And overall, it has been demonstrated from time to time that prevention is better than cure. And therefore, when pest risks are identified early, it is more rewarding to prevent them than the interventions that normally come later on when it comes to pest management, when the pest has invaded. In this particular curve, you see the area of the pest invasion increasing over time. And when the pest has been introduced over time, there is different stages. We have the prevention stage, we have the eradication stage, the containment, and asset-based protection. We are concentrating on the prevention stage. On the prevention stage, and in this aspect, we are looking at any measures that will be able to be implemented at the very beginning when the pest has not invaded this particular space, be it an area of concern, it can be a country or a region. Over time, when an invasion occurs, the pest that has been identified when introduced, it starts with small numbers as localized population, but as time goes by, we are likely, if we don't eradicate the pest, we are likely to reach levels where the pest has invaded critical ecosystems, and therefore we are not able to manage them. So in this particular generalized pest invasion curve is to demonstrate that prevention indeed more rewarding. There's a high cost benefits than if we wait to manage the pest when it's already invaded the systems. Under the plant-wise program, we are really basing our interventions under this pest invasion curve. We are having measures to be able to identify pest risks, to be able to undertake risk analysis of these pests, to institute import control if they are facilitated through pathways, develop contingency plans, and be able to manage the pest risks before they occur. And we believe that if communities are capacity built to be able to identify these pest risks, be able to manage these risks over time, monitor them, institute contingency measures, including surveillance and eradication programs, we are able to keep some of these high-risk pests out of reach of key areas, and therefore protect them and reduce the impact of this particular risk. Now, in our interventions, we have activities that target prevention, identification, risk management, and also pest risk management, like IPM, biological control, farmer training in readiness of invasion. So some of the initiatives that we have undertaken in the Asian region include supporting risk assessment based on biosecurity in the region in Asia. And some of these initiatives have included identify the pest risks. This is through horizon scanning, assessing their risk of introduction, their impact, establishing a pest risk register. This is a list of identified pest risks in a particular country or a region, and then strengthening the national plant protection organizations to be able to undertake systematic pest risk assessment and to increase their overall awareness in terms of, where do they get information, what tools do they use to undertake a pest risk analysis, and what do they need to undertake in terms of contingency measures in readiness for invasion. Usually it's very difficult to institute some of these measures because most countries are used to pest management, that is asset-based management, when the pest has already invaded the area rather than before. So in this particular case, we are preempting introduction and therefore instituting measures and that will keep the pest away from the countries of interest. The countries where we have undertaken some of these initiatives in South Asia include India, Nepal, Sri Lanka, and Bangladesh. And in this particular picture, you see a horizon scanning workshop to identify some of these pest risks in Nepal. We also see a number of participants, basically from the National Plant Protection Organizations, but beyond that, other stakeholders that support the NPPOs in their pest risk activities. We have been able to also include some of these approaches in Bangladesh, Nepal, Sri Lanka, and mostly we have focused on arthropod pests and diseases, and we'll be moving to how to prioritize the weed species that also can be able to be introduced into the different areas and threaten the agriculture and economy of these countries. So some of the pictures you see here include pest risk analysis of prioritized pests. Once you identify these pest risks, you need to undertake a thorough risk analysis, identify some of the pathways that can introduce them, and also institute measures that can help to mitigate the risks. How do we undertake the horizon scanning? By definition, this is a systematic examination of potential threats and opportunities that can aid their invasion in a particular area. So in this aspect, we are talking of a country, like the case of Sri Lanka or Bangladesh. So identify all pests that can be able to get introduced in these countries. Remember, the pests are not yet there, but they can be able to come through different pathways. And also scoring the risks. And when scoring the risk, we are using a number of tools. First, we are using the Kabi Horizon Scanning tool to be able to identify these potential risks. We are using the Kabi Compendium datasheets to be able to learn more about these pests from wherever they exist, and also to identify the different pathways, the impact and the climate. Remember, if a pest does not have any impact on the country that is at risk, then of course the risk reduces and it's not prioritized. We are using the Kabi Horizon Scanning tool to be able to identify this. And I think some of you might be aware of this tool. There's a free version and of course premium version, and it supports the countries to, at a click of a button, be able to identify some of the pests that are of high risk of introduction, and therefore reviewing them, also using the Kabi PRA tool to be able to ascertain their risk. Currently, we use this output from the Horizon Scanning tool to be able to prioritize the high-risk pests so that we can be able to institute measures to prevent them. We can be able to plan to undertake surveillance for early detection, and we can be able to institute measures, either import control, to make sure that the identified pathways do not pose any risk of their introduction. I will not go into the details of scoring risk, but it's important that to underscore the fact that you can, through the horizon scanning tool, you can be able to even identify maybe hundreds of pests, but you need to prioritize these pests based on their risks, and therefore risk scoring is important to help you identify which pests are likely to enter, which pests are likely to establish in a specific area, what is the potential economic impact, and what is the environmental impact. Remember, a pest that are high risk is likely to be able to enter, to establish, and to cause economic impact. But that's not to say that the pest that will end up with a low risk cannot be able to enter the specific area at risk. It can, but then we have limited resources, and therefore we need to focus on the high risk pests. Once you identify the high risk pests, you can, through the National Plant Protection Organization and the other stakeholders, establish a pest risk register.
Another
In this
3
register,
minutes,
we keep a record of all the
Another
pests
3 minutes,
that
please.
have been prioritized. For instance, they are of high risk of entry and high risk of impact if they enter into the country, and therefore you want to start monitoring them. You want to review the scores. Sometimes the scores become higher. Sometimes the pest is finally even detected in the area of focus, and therefore this pest risk register is an important tool that the NPPO establishes to be able to keep a record of high risk pests, but also monitor them and also allocate resources for their surveillance. So far, we have established the pest risk register in Bangladesh, and also soon we are establishing in Nepal and Sri Lanka, and keep on monitoring them to be able to work with the NPPO to ensure that resources are allocated for the monitoring, but also for any action that may be required on this particular prioritized pest. Pest risk analysis is quite essential for you before you actually, as you monitor the pest risk register, you need also to assess what pathways are these pests likely to come through. So evaluating the biological and scientific and economic evidence to determine whether the pests need to be regulated or to strengthen the phytosanitary measure. It could be a review of an import condition or the like, so that we can be able to institute measures to mitigate the risk of these pests entering into the countries. It must be science-based, and therefore we need to be careful not to institute measures on pests that are of little risk into the country, of low risk into the countries that we are looking at. This is just a flow of the pest-initiated PRA. Remember, when you're undertaking pest risk analysis, you can either look at the pathways and identify the pests that can follow that pathway, or once you have identified pests of high risk, like the case of the pest in the pest risk register, you can institute pest-initiated PRA, and it can help you to identify the pathways and also help you to identify mitigation measures that will help to manage that particular pest risk. So this is our PRA tool that helps countries to be able to undertake pest risk analysis, either pest-initiated or pathway. In this case, this is for the pest-initiated PRA. We've also really undertaken capacity building into the countries that we are involved in the Plant-Wise Plus project. So capacity building, the training workshops, or real-time PRA activities. For instance, in this page here, you can see tutor absoluta, pest risk analysis on that, and some of the countries where we have capacity built PRA. This is Bangladesh, the NPPO, and also the facilitators. We've also had projects with the FAO to be able to undertake capacity building on risk assessment for seed pests. We know pests can be a pathway for
So, thank you very much, Madam. And the questions will not be there because it is a recorded statement only. And we thank Dr. Mary for having given us the insights of strengthening of biosecurity. Now I call upon Professor Dr. Rakesh Pandey, Emeritus Scientist, CMAP. He is going to deliver PR Verma the next award lecture. Please. 15 minutes, sir.
Good afternoon. So, tonight we are having a very good hall, and this entity unique hall. I think wherever we go here, I think the issue for essential. We are going to for something. So, Let it go. Otherwise. Okay. So first of all, I would like to give my sincere tribute to Professor P.R. Verma, sir, to whom I met so many times before 2015, whenever SMPP conference is going on, and most of the time Professor P.R. Verma is there. So at least I personally met Professor P.R. Verma, and who mainly work on the albigo candida, which help in developing a successful technique for the red seed germ plant. And most important part of his research that he has license of the world first white rust resistant Brassica cultivar in Canada. So this is the major work, Professor Verma. So my sincere tribute to Professor Verma. Where is the slide? How can I move? From here? Yeah. From here I can move. Oh. Right. Okay. So before coming on my topic for the discussion, I am highly and sincerely grateful to SMPP students and judging committee who has selected me for this memorial award and for providing me this opportunity to discuss some of my idea with all of you, and organizing committee of the 5th ISMPP International Conference in this beautiful complex of the MT University, and my special acknowledgement to Professor Salini Singh. Now, I'm going to talk about the green strategy and innovations for plant and soil health management, a climate resilient approach. So, because whatever we are going to talk, at least climate is playing major role. And if I'm not talking about my institute, it will be injustice, where I was 42 years as a scientist. So if you see the medicinal plants in India, every type of land, in every season, even higher pH more than 9, more than 10, we can go medicinal plants. For example, in dry land, we can grow the Pomarosa, vetiver, lemongrass, ossimum. Then flood-prone area, we can grow the vetiver. Then primary and secondary salinity, when we are having higher pH, even more than 8, more than 9, we can grow some of the aromatic plants. Then orchard and plantation, we can have patchouli, then eroded and degraded wasteland, you can say. There we can go Pomarosa, lemongrass, and even hilly area. There are some of the medicinal plants that we can grow in the hilly area. And these are some of the medicine plant. Now I am not going to tell you, but this is the picture of medicine plant. Now farmers are cultivating and they are getting very good income. Even if they are cultivating vetiver in 9 months from 1 hectare of land, they get 1.5 lakhs rupees profit. If you see the cost benefit ratio. So these are the different medicines like ashwagandha, opium poppy, Golfia serpentina, then anti-malaria drugs, Artemisia annua, then isabgol and this curcuma. And during the COVID, you see the Calmeg, Andrographis paniculata is the lowest one. Here we have developed a new formulation for the immunomodulator. Then these are the mentha, I think you know very well, satawan, and these are the aromatic grasses. And these are the different medicine plant. Generally, we are using the molecules, oil and extract. And there are some herbal drugs our institute has made, like this. You can see variety of the products are available. There is no any synthetic material, but these are based on the plant material only. So these are the institute's products. And now I'm coming, this is another product which we have developed in CMAP. A tea we have developed by working on the Caenorhabditis elegans. This is anti-aging and cognitive booster tea. This tea we have licensed to the Biome Pharmaceutical in 15 lakhs rupees. So by working on the simple nematode, we can develop a very good herbal product and which is certified also. Now, if we are talking about the climate resilient, its ability of communities and ecosystem to anticipate, accommodate, or recover from the climate-related distress from plant and soil health. Because these climate change, there are two regions. One is the natural, another is the anthropogenic. So natural, we can't have any control, but anthropogenic, these are the major causes of the greenhouse gases. And especially if you see the deforestation and urbanization, a lot of deforestation and urbanization is going on. Even if you see the nearby Lucknow, under 50 kilometers, people are making the urbanization, no agricultural land. So they are utilizing all agriculture land. So ultimately, there will be the change in the temperature, and when human activity like burning of the fossil fuel, coal, oil and gas, electricity, heat, these all are responsible for the increasing the temperature and causing the global warming. And here, what are the major problems? When there is a climate change, especially being a plant pathologist, we have to talk that there is more severity of the plant diseases. There are so many impacts, but the major impact is that more diseases are there and their severity is continuously increasing. And here you see, I have given two examples. So when we are having optimal climate scenario and there is climate change. Now there is no need to describe each and everything. One can imagine what will be happen even in future. And here I have shown you that if there is a optimal condition, we are having beneficial microbes. But when we are using the chemical fertilizer, even chemical pesticide, even chemical nematicide, we are going to change the soil microbiome. Whole of the soil microbiome, whole of the useful microbes, we are going to change. So we have to take care that when you are using huge pesticide, chemical fertilizer, then you are going to decrease the soil health. And soil health is very important for the plant health. So this is our prime duty, how to maintain the soil health. If you see here how I have collaborated, if you see the soil health, physical, texture, water holding capacity, pH, these all, they are responsible for the soil health. If we are going to kill the useful microbes, if we are going to use the pesticide, we are going to change the soil microbiome. Ultimately, it has impact on the plant health. If plant is not healthy, then we can imagine a good health of the human. So ultimately, human health will be also affected. Here, one of the very good example I have given you, like European Commission in June 2022, they told that 50% reduction in plant production and hazardous chemical. But when I was studying last 10 years, even after 22, whatever rate they have given that it will decrease, but you see it is continuous increasing. If you go to the literature, if you go to the data, continuous use of the chemical is increasing. It is not decreasing. So ultimately, this will impact on the disease. Like here, I have shown some of the diseases on the plant. And here in this picture, I have tried to convey the message that how these four chemicals, different type of chemical pesticide, going to have impact on the useful microbes and severity of the disease that impact the plant health and human health. And there is a tug of war between the pest, pathogen and host. Generally what we need such type of the crops, healthy crops. But what happens generally, we are having the diseased plant. Either it is nematode, either it is virus, either it is bacteria, either it is fungi. Continuous, there is a severity of the diseases increasing. And people tell that it is temperature increases 1 degree centigrade. But somewhere it is 1.5, somewhere people are writing 1.9 degrees centigrade. The continuous temperature is increasing. But what plant pathologists we are doing? We are playing with these three: pathogen, host, and environment. Just we are manipulating, either we are using microbiome, either soil microbiome or philosphere microbiome, whatever we are using, we are just playing with these three factors. And if you are manipulating these three, you can easily manage the plant disease. So whatever we are doing with these three. Here, these are the diseases. I have told you, we all are aware about these different diseases, and there is no need to take. But one thing I would like to convey here, there is a yellowing in the grasses. This yellowing in the grasses at the corner, I am seeing last 20 years. Being a plant pathologist, I am telling you, all plant pathologists, this disease, yellowing of the aromatic grasses, still yellowing is there. People has not able to manage this yellowing of the aromatic grasses last 20 years, I am telling you. I am seeing this disease not only in the UP, in Lucknow, even in Northeast, I have observed this yellowing. But still there is no cure of the disease. These are the challenges. I am not going to read it, and you know very well. And here these challenges, what economics, environmental health, and whatever technology we will develop, we have to take care of the environment, health, and economics, and especially farmers. Microbiome, no need to tell. Everyone knows very well how it is going to impact on the plant and human health. And these are the different omics technology. Huge number of the omics technology now available, and how we can apply these omics technology to manage the plant pathogen and to make the farmers. And here, this major strategy for the management of diseases, like biological consortia and disease resistance. In disease resistance, now people are going to CRISPR technology. Now, at least in January 2026, we have two varieties of the basmati through CRISPR technology. But consortia, I have showing you in the middle that in CSR CMAP, we have developed that Calicarpa macrophylla, that we have isolated calicarpinine, and then mycorrhiza and some useful bacteria, and we make a consortia, and we have used in the medicinal plant, and they have got very good secondary metabolite, enhancement of the secondary metabolite. And whatever technology we have discussed, it is reached to the farmer field. I'm not going to tell you that these technology, what we have developed in CMAP, it is in our lab or it is in our research paper. No, these all technology have reached to the farmer's field. So that is the beauty of our science. No need to tell about this genome editing. People are doing, and still some new variety came in India. And especially on this microbiome, we are working, and we have got very good success. And these are the innovative work. Ultimately, these are the useful characteristics of the microbes which are present in the soil or which are present in the philosphere. And by utilizing them, by characterizing them, we can develop some of the new formulation or new technology. Then same thing, how we can manage the disease in the plant. In the right hand, I have shown you that if we are going to follow these technology, ultimately we can increase the plant growth and yield and decrease the plant diseases. Here, same thing, triangle, I have shown you the host, environment, and microbes. And even though if we are working on different type of microbiome, we are playing only these three triangles. Even human attraction is also there. But whatever microbiome or different omics technology we are using, we are playing only these three: host, pathogen, and microbes. Here about the nematode, you know, nematode is the major problem. Generally, people ignore it, but it is just like a cancer. I don't like to show you the figure, but there is once nematode enter inside the root, it hurdles the vascular bundle, and whatever nutrient or water or other uptake material is coming to the stem, it is not coming, and they disturb a lot, and secondary metabolite is also going to be affected, and we have proved it. So how we can manage using the different nematophagous fungi, and these are the direct impact of the trichoderma. I think the Dr. H.V. Singh is here. We learned trichoderma from him. So how trichoderma is useful, I'm not going to describe because we all are aware about the trichoderma. But one thing I tell you, when we use trichoderma, we manage the nematode problem in the menthar, it means mint. So we use, and this trichoderma reaches to the farmer field. And this is the different characteristic of the trichoderma, need not to tell you, because, and this slide I want to tell you in 2 minutes. This slide, I want to tell you that we have gone to the farmer field, because ultimately what technology we are developing, it is not going to the farmer, there is no use. So whatever technology we have developed, either it is based on trichoderma, either it is the consortia, either it is based on some of the useful bacteria, that technology has gone to the farmer field and it has been appreciated by the farmer, not appreciated by the authority. If my research can bring a smile on the poor farmer, then that is my real science. So I believe in such type of the science. With this, I think this is some of the work we carried out. I'm not showing more of my work, but 1 or 2 slides I am showing you how we use and how we manage the nematode problem in medicinematic plant. One more slide is here. These are the expectation and these are the benefit. No need to tell you because you all are about the benefit of this thing. And nowadays people are talking nano fertilizer. But I have gone in the field. These nano fertilizer or nano area I have visited in the northeast and the other part of the country. Nano area is still not working. I'm not convinced with the nano area. You may be convinced, but I'm not convinced with the nano area. Then my last slide, sir. So great thing always comes. This is my group, because without them, whatever I'm talking, I cannot talk to you. This credit goes to my student, not to me. And this is my Mano Park of my institute. You see, if students are visiting, they know which plant is used by which part of the body. So this is one of the beautiful park of my institute. And if somebody get time, they should visit there. And thank you very much. I will be happy to answer any queries.
So thank you, Dr. Rakesh Pandey, Emeritus Scientist from CIMAP, for delivering very interesting lecture of for P.R. Verma Award lecture. This is, give a big applause to him. I welcome Professor Dr. U.C. Lavanya from the university, from CIMAP. Earlier he has worked at CIMAP. I welcome him. And he's a well-known cytogenetist and biotechnologist. Thank you. Now I call upon Dr. Muhammad Akram. He is going to speak. He is from Division of Crop Protection, ICAR, Indian Institute of Pulses Research, Kanpur, on Begoma virus complex in pulse cropping systems, diversity, epidemiology, and crop feed interaction. 15 minutes, sir.
Respected chairs, distinguished delegates. Pulses are very important for 2 reasons. One is that they have the nutritional quality, means high protein, and then they are able to fix the atmospheric nitrogen. So keep them healthy, it is our priority. There are many challenges. One of them are the bugemoviruses. So I will be talking only 2 things. One is that the bugemoviruses associated with yellow mosaic disease is this #1 disease in pulses, and since the 1940s it is still continued and it's a #1 disease. Another one is that bugemoviruses causing leaf curl kind of disease. If you see taxonomically, this group belongs from the Geminiviridae family, and so far it is largest one. More than 460 species are there in this particular group of viruses. And then they are basically the DNA virus, single-stranded circular DNA virus, and they are divided into 2 parts, bipartite and monopartite. Means there are, if the DNA A and DNA B are together, so they are bipartite. If it is only the DNA A, so it is monopartite. And then the particle is geminite, twins kind of particles are there. And they are exclusively transmitted by whitefly. And then if you see the genes, in DNA A about 6 to 7 genes, and DNA B has only 2 genes. They are having all sort of functioning in that. If you see that the groupings of these viruses, so all bugemoviruses fall into these 4 groups, and then under them the 3 one is the legumoviruses. They are exclusively infecting the legume host. Besides this, they are also having some satellites like alpha, beta, delta. So these are the satellite DNA molecules. They are also associated And then because of the association of these, there may be sometimes the high disease or sometimes there may be a low disease. That's why I'm calling it the Bugamovirus complex, because there are beta satellites, there are alpha satellites, there are delta satellites, and then DNA A, DNA B, and then combination of these. And then if you see the how we are saying they are the 400, more than 400 species, because of the there is a criteria based on the full DNA sequences. If it is less than 91%, it is different species. If it is more than 91% matching with any known virus, it is the same species, besides the other biological parameters. Now it is very interesting. In the morning itself, our president ISPRD said that seed-borne kind of things he said, and then there is a still unresolved mystery. So, and then he was saying only the mung bean yellow mosaic virus. But you know, in the yellow mosaic disease, they are caused by about 13 different viruses. Of these 13, 9 are present in India and 8 we are having in our lab. So we are having the clones of these viruses in our lab. 8 are present in that. And recently we characterized the first one, that is butterfly P yellow mosaic virus. So all these 9 viruses are causing yellow mosaic symptoms in the pulses. And then these are the hosts. More than 6-7 plant crops or crops have been affected by these viruses. But 3, Horsegram, mung bean yellow mosaic India, and mung bean yellow mosaic, they are most prominent, more prevalent, and distributed almost all part of the country. And then these are the viruses causing leaf curl kind of symptoms. And most of them, they have been reported from tomato, but they are also visitor. We are calling them, they are the visitor to the pulses also, but they are not causing much of disease, except that French bean leaf curl virus, it is from the French bean. And then these are the satellites, they have been reported in pulse hosts. Earlier, we were thinking that legumoviruses only infect the legumes, but it is not like that. There are reports that they are infecting tomato also. Even we report that even lentil also having the infection of mung bean yellow mosaic India virus, but they are only the isolated plants. And then since 1972 and 74, 77, there were about 75 weeds. They have been reported showing the kind of yellow mosaic symptoms. But till 1940, 2014, there was no research on these particular aspects. And now it is confirmed that there are many weeds, they are harboring the mung bean yellow mosaic virus or the horsegram or means YMD causing virus. And this information is very important because the primary source of inoculum goes from these hosts to the main cultivated crops. So we also identified these weeds, they are harboring the virus, even they are not showing any kind of symptom. And then this is the butterfly one. This aparajita is we identified that yellow mosaic symptoms were there, and then when we saw the virus, so there are 2 viruses. Even mung bean yellow mosaic India virus also there in that. And then in this wild pigeon pea also they are harboring mung bean yellow mosaic India virus and then Nicotia yellow mosaic virus. So this pigeon pea also having the mung bean yellow mosaic disease or this yellow mosaic disease. Earlier it was not there in the pigeon pea, but now it is coming in the pigeon pea also. And then the important point is that there is a mixed infection also. So we collected these 2 samples of Vignes stipulaceae and moth bean, and we identified that 3 viruses are present. All the genomes of 3 viruses we get from these. And then based on the more than 250 samples collected from 38 different locations from the country, more than 50% samples showing the mixed infection of these viruses. So, and because of this mixed infection or the individual infection, sometimes the disease is more or sometimes the disease is less. We also develop one this countrywide distribution map, and it is available at our ISPR website. If anybody can go and check it, and then at which locations, which virus is present in which crop or which host, this information is available, and that based on this whole analysis. And now coming to the diagnostics. So for ELISA, detection is not working in that. And then since the yellow mosaic disease is common, symptom-based diagnosis is not possible. So we have developed the single PCR detection techniques for all these viruses, all 8 viruses, and then based on the specific primer. So no need to go for the sequencing, just to use the specific primer, and you may say that which virus is causing the disease in that one. We also developed the multiplexing of 4 viruses, and then even a single reactions based on the amplicon size, we can detect any of the viruses present in the sample. And this is the golden question, that whether it is seed transmission or not. Generally, big gamma viruses are not seed transmitted, not seed borne. And then there are reports that somebody has say it is transmitted, then subsequent paper came, it is not transmitted. This happened 3 times and 4 times in that. So I'm not going to say much in detail. See that this first one, 1934, they say it is seed borne. But again in 1955, they say it is not seed borne. Another paper came in 2016, say this virus is seed borne. And then again, then 2017, they say it is not seed borne. And then see the paper also. And then in India, based on this YMD, this Mahatma, our president, first time he raised this question. And then he said that the seeds parts having the virus. Yes, in all the seeds parts, virus is there. We agree. But they said that the symptoms are not coming. And then until the symptoms will not come, how we can say it is seed transmitted? So still it is debatable this. We also conducted experiments on that. We took all parts of this, taking the one susceptible and one resistant cultivars, and then go for the growing test. And then if you grow in control conditions, there is no chance of whitefly. Definitely you will not get any symptoms. Even the seeds were collected from the infected plants, seeds having the positive in the PCR. And if you grow the same variety outside, you will get the disease. And then we are trying to see that how it is going on and what is the reason for that. So we detected that in all parts of the sample using a small fragment of PCR, and it is found in all parts of that, even resistant and susceptible one. But when we try to amplify the full genome, the complete DNA, so plumule, there is no detection of the virus. It means that something is there which is chopping this virus or degrading the virus. Then after that, they are not going to be recirculating, or they are not going to be multiplying. So because of all these experimentations, we concluded it is not seed transmitted. But still there is a need to work on that. There are many management options. The only management option is workable. It is the resistant cultivars. And then we are continuously developing the resistant cultivars. The only thing is that we have to grow the resistant cultivar in a region where it is being released. If it is going beyond different region and different season, there are chances of getting the disease. So there is no absolute kind of resistance is there. But they are the resistant cultivars which we develop in mung bean and urd bean, basically. Of course, we go for the management of vector. But if you spraying only your field, and then most of the insecticide are systemic in nature. So whitefly first will feed, and then it's going to be died. But meanwhile, it can transfer the virus. So we conducted experiments in that. We spray 3, 4, 5 times. Nothing will happen. Disease will not going to be reduced if you are not spraying the larger area. So this is our conclusion. But anyhow, we have to have seed treatment and then to spray, then definitely the yield will enhance. People are working on the transgenic. There are many viruses they have tried. So, but still nobody has tried on the YMD viruses on that direction. And then people are also trying to characterize the gene for resistance and susceptibility, because once we are having these genes characterized, we will use for the genome editing or for this one. So still it is in infancy, and then still we don't have any genes characterized whether the resistant or susceptible in pulses against these viruses. Durable resistance is very important, and then continuous breeding is again very important. Then characterization of resistant gene and susceptibilities should be in priority area. And then of course rapid diagnostics is still it is required for surveillance. And then chemicals. I believe that if we use the repellent kind of chemicals, so that will be more effective than the killing kind of this one, because in repellent the whitefly will not feed, and if will not feed it, definitely the virus will not be transmitted. Thank you very much.
Any questions, please? Yeah.
Okay, Dr. Akram, now nice presentation. Really, actually, white fly, it's play a very important role for the severity of the disease. So now one of my question is, now you are working on white fly. Have you idea regarding the any equipment for the measuring of white fly population?
Equipment is very simple.
No, it's very simple.
Population,
It's okay. Mechanical and all these things is okay. But just like you recall regarding the disease incidence by collection of the sports by sport traps. Sport trap is one of the very common equipment. Regarding this one,
yeah, we are having yellow trap.
Yellow.
You just put in the yellow sticky trap. You just place in the field for 24 hours and then see the counting. White fly will come and then trap it. Because
Right. For actual counting, I think now yellow trap or mechanical जो भी हैं आपके, I think it is not follows for the epidemiological work. For the epidemiological work, now need any equipments.
Yeah,
Any equipments. Just if you recall, now one of the I don't know about the, again some of the Austria people now develop the pole-like structures, and these pole-like structures now generally follow for especially for the white fly. I doesn't know about the actual mechanism for these activities, but now specific equipment just like sport traps.
It is simply the trap. We are having the yellow trap, but even we are having the one small box where you can fit a tubes, and because of the phototoxic this one white fly automatically
you can count the actual population
yeah
yellow trap
easily with the naked eye.
In case of the cotton or in case of the soybean, now the population is too much more. So in this case, it is possible through the yellow trap.
Yeah, it is again possible. They can give you an idea how much, or you simply go for the trapping. Means there is a black box. You put in the black box on that. Give some time. White fly come into on the glass. Take a picture and then count it. This is another option. So already these things are there, but they will not count accurate.
Scientifically, actually, I want to scientifically.
They will not count accurate, but they can give you right.
But anyhow, he wants accurate methodology, and that you can discuss with him.
Yeah.
And what is the role of satellite molecules?
So they enhance sometimes the disease severity. Satellite molecules.
But it is not clear.
No, it is still nobody has estimated proper.
Okay, thank you, Dr. Akram sir, for a nice presentation. And Begoma virus is very important one in the case of pulses. Okay, thank you. And now I call upon Dr. Daisy Vasantarai. She is the Joint Director, School of Crop Resistance Systems Research, ICR National Institute of Biotech Management, Baranda, Raipur. And she is going to speak on innovative breeding techniques for enhanced resistance to diseases in wheat and rice. 15 minutes, ma'am.
Thank you very much, sir. Good afternoon, everybody. I am Dr. Daisy Vasandrai from ICR NIPSM Raipur, and will be talking on innovative breeding techniques for enhancing resistance to diseases in wheat and rice. Why this is needed? We all know that when we see in 1950s, we were importing a lot of food grains, and that was also very spurious and not up to the mark. And after that, with the introduction of Norrin-10 and DG genes, we could identify new varieties, and we could find that now we are self-sufficient, and rather we are exporting wheat and rice to other countries also. This is because of not only because of breeders, but because we are breeding for disease resistance also. And still, although we are doing a lot of work, but still there is 20 to 40% of potential crop yield losses is lost annually due to these insect pests and diseases across major food crops worldwide. And these losses, they directly threaten food security, farmer livelihood, and sustainable agriculture, especially in the developing countries. These pictures are, I think, I'm a breeder basically, and this picture is
known to every pathologist that whenever there is host, pathogen, and environment, there will be a disease, or the interaction of these three are very important. And everybody must be knowing about Dr. R.H. Painter. He is known as the father of host plant resistance, and nobody can forget the concept of floor hypothesis, in which wherever there is a virulence gene in the pathogen, there is resistance gene in the host, and that interaction will give rise to resistance. And this Floor's hypothesis of host resistance gene and virulence gene in the pathogen has led to the plant immunity system, which is the zigzag model of plant pathogen interaction, which is nowadays it is called as the central dogma of plant pathogen. And in this case, we know that the plant immune system, it carries two interconnected tires of resistance. These are sensing molecules outside the cell, and the other one is the sensing molecules inside the cell. And both the systems, they sense the pathogen and respond by activating defenses in the infected cells. So first one is the pathogen pattern triggered immunity, that is the PTI. And this PTI is activated. We all know that this PTI is activated when plants detect pathogen associated molecular patterns, such as flagellin or chitin. And this PAMP is further recognized by the pattern recognition receptors, PRR, of the host, and it triggers a defense response. This is the first immunity system, that is PTI, when the pathogen enters inside the cell of the plant. And the second one is the effector triggered immunity, which is very important nowadays. That is the second tire is governed by the resistance genes product. And this effect, ETI, is activated by the presence of specific pathogen effectors. And some pathogen, they bypass PTI by delivering effector molecules into the plant cell. And then the plant resistance protein, they recognize these effector molecules and result in stronger response, which includes a hypersensitive cell response, cell death also. And this layer of immunity is referred to as the gene for gene interaction between plant resistance gene and the pathogen effectors. Here we can see the first figure is about PTI, that is PAMP triggered immunity. And the second one is the effector triggered immunity, which is very important as far as breeder is concerned, because the pathogen, it sends its effector molecule inside the cell, and the host resistance gene, which is NBS LRR gene, they interact with the effector molecules, and further it gives signals for the defense response, and there will be salicylic acid or reactive oxygen species hypersensitive response. It gets activated and the plant shows resistance to the host. If we see that disease resistance, a lot of varieties have been developed with varying degree of resistance is also there. And the two broad categories, which we all know, that there is a race non-specific resistance, which is broad spectrum, durable, and mostly polygenic. And there is race specific resistance, which is controlled by major genes. And it is based on the gene for gene concept. It triggers the effector triggered immunity, that is ETI system, and leads to hypersensitive response. And there is one more type of resistance, that is the non-host resistance. And this non-host resistance, we are working on what are the factors which are responsible for non-host resistance. And we have identified in NIBSM, we have identified some factors which are responsible, and we are validating those factors. These are the transcription factors which are responsible. So we go the classical method of breeding, because this topic is for breeding methodology, how we are using the pathological concept in the breeding part. And if we see the method of breeding, we are mostly using selection. Then varieties are introduced, some mutations are they were done. And on the basis of mutation, new varieties were developed. Then hybridization. In hybridization, there are different methods: pedigree method, backcross method, and single seed descent method. But there are limitations of this conventional breeding program, because it is time intensive, linkage drag problem is there, and there is narrow genetic diversity also. And if we see the breeder's favorite equation, this is the RT, that is the genetic gain over time is directly proportional to the selection intensity, selection accuracy, and genetic variance. Wherever we can have selection accuracy, only then we can have more genetic gain, and it is inversely proportional to the years per cycle. So we have to reduce the year of breeding, and we have to increase the selection intensity, accuracy, as well as the genetic variability. Only then we can have more genetic gain. This is the favorite equation of all the breeders, and we are working around only these type of equations. So if it's then after the when the tissue culture era came, then wide hybridization, that means genes from wide crops, they were wild species, they were transferred using tissue culture methods, homoclonal variations, somatic hybridization, meristic culture methods. These were all tissue culture based techniques, and lot of varieties were developed because of the resistance genes, LR genes, YR genes, PM genes. They were incorporated, likewise in case of rice, PI genes for blast and XA genes for Xanthomonas oryzae species. So these were another era. And after that, when the DNA markers were identified, then and the whole genome sequencing was discovered. Then after that, a new era of plant breeding started. That is from that marker-assisted selection, marker-assisted backcross breeding, marker-assisted pyramiding, and then because of we know the sequences of the genes also, then we can go for genome editing of the susceptible genes, and even the promoter regions, they are being regulated. And then another area is transgenics and RNAi interference, but although these are not approved in Indian conditions, so we will not be discussing. Then genomic selections, genome-wide association studies, DH, speed breeding for reducing the time, and high throughput phenotyping, high throughput genotyping, and AI-assisted based breeding. So these are some of the most important parts which we will be working. And in case of, if we start with the marker-assisted selection, these uses DNA markers which are linked to specific genes or QTLs controlling desirable traits, and this allows to identify and select favorable plants at early stages of the genome based on their genotypes. And in this case, steps mainly are that we have to identify the markers, then we have to validate the markers, utilize those markers, and simultaneous marker development again is being done. Then there are QTLs have been identified for specific gene resistance, for resistance. So all these markers, they are being used for identification of genes which are present in the varieties and further transferring the genes. So if we see the conventional methods, we cross two parents, F2 is being screened under glass house conditions. But with the help of marker-assisted selection, we can just, by looking the bands which are obtained in F2 condition, on the basis of markers, we can identify the plants, resistant plants or susceptible plants, and then we can further progress those plants. So, and then these marker-assisted backcross breeding was started. And in this case, again, the resistance gene can be transferred into the susceptible parent, and which is a high elite parent. And on the basis of foreground, background, and recombinant selection, we can identify the new genes genes in that. Then marker-assisted pyramiding is being done, in which two genes can be incorporated in the one species using the markers for particular genes. So there are many examples now which have been, many varieties have been released because of these marker-assisted selections, marker-assisted backcross breeding, and marker assisted pyramiding. So these are some of the lines which in case of rice and wheat, where these YR15, YR17, YR10 genes, they have been combined. Likewise, X821, 53, these are some of the genes examples. And these varieties are in the field also, and farmers, they are growing these varieties in a big way. The second part is the most revolutionized is the genome editing, which breeders are using. And this is the genome editing. It was has been selected as the method of the 2011 method of the year, and CRISPR-Cas especially it is the breakthrough of the year. And in this case, again, there is it is the ability to modify the DNA sequence of the target organism. And it is a type of genetic engineering in which DNA is inserted, replaced, or removed from a genome using artificially engineered nucleases, or which is called as the molecular scissors. And these nucleases, they create specific double strand breaks at desired locations. And then after joining, homologous joining or non-homologous end joining, there are some deletions, additions, and duplications. And then that particular gene, it is being edited, and the product which is formed by that particular gene is changed. And the general protocol, if you see that, first of all, we have to select the genomic target, then design guide RNA, then assemble of Cas9 and guide RNA construct. Then these are delivered to the plants, then plants are regenerated, and further we can identify the gene edited plants from the even this gene editing can be done on these susceptible genes. Most of the sweet genes, they are susceptible, causing susceptibility, and if we disrupt the susceptibility genes using genome editing method, so we can have resistance. And likewise, there are many broad spectrum resistance can be obtained by editing genes S5H mutants. Then these are some of the examples where edited genes have been edited due to CRISPR-Cas method, MLO genes, EDR1 gene, DMR6 gene, and there are the sweet genes. And even if we edit the promoters also, then also we can have resistance, which is promoters which are involved in the but not using we are not editing the gene, rather we can edit the promoters also for resistance. So these are some of the examples where promoters have been used for gene editing. Then two varieties, we all know that they have been released because of, although the disease resistance has been not used, but the drought and cell entity has been used, these two varieties. Transgenic approaches, we all know that, but since we are not working much on this RNA interference because it is banned in SDN 3 and 4, it's not. Another method which is being used is a genomic selection method, and it is again a specialized form of marker-assisted selection in which we use the genome-wide markers. On the basis of training population, we get the genome estimated breeding values, and on that basis, we can identify new genotypes. And further integrated genomic selection, in which we are using the speed breeding also, which this genomic selection is integrated with the speed breeding so that we can have in less time, we can get new varieties. Speed breeding, we all know that by reducing light intensity, light quality, we can, and then day length and temperature, in whatever we are getting in 6 generations, we can have 4 to 5 generations we can have in 1 year. And with this, we can reduce the time of breeding. Then high throughput phenotyping also, we can have very good phenotyping. And these are the, if we integrate data from genomics, transcriptomics, proteomics, and metabolomics, and combining the high throughput data, using bioinformatics as well as machine learning algorithm, we can have new traits, we can accelerate the breeding situation. Thank you very much. Breeding for disease.
Thank you, madam. Dr. Daisy, madam, has delivered very interesting lecture. And anybody, if have got any questions, you can discuss with her later. But the most difficult thing is in innovative breeding for enhanced resistance is rapid evolution of plant pathogens and limited availability of germplasm. This is both are difficult to achieve together. Therefore, in spite of all this, she has presented very well in this. Now I thank you very much, madam, on behalf of ISMPP and Amity University. Now coming to, instead of Dr. Anil Kotastane, and a new speaker has come into the picture, Dr. Deepak Singh from CSIH Lucknow. 15 minutes, sir. लगाए दीपक सिंह
ये वाला start.
Uh, Good afternoon, everyone. Myself, Dr. Deepak Singh. I am working as a head division of crop protection, ICRC, Lucknow. My topic is the recent advances in the management of the post-harvest diseases in mango. Actually, in case of the mango, now we are facing a lot of problem. First problem is early morning we are discussing with our colleagues. Wilt is a major problem in the mango. Okay, in our this particular area. After that, one other thing is blossom blight. Blossom blight is another problem, and most important thing is the reduction of the this pollinator population. Actually, due to the spray of the huge pesticide in mango orchard, the decline of the pest in this particular population of the pollinator is decline very fastly. Actually, we are seeing good flowering in case of the mango, but fruit set is very less. This is indicating how we can revive our this particular population in mango orchard or ecosystem also. And now we are discussing about the post-harvest diseases. About that is very important anthracnose as well as the stem introt. But now we are focusing only here on the anthracnose disease. In post-harvest diseases, near about the 20 to 40% losses has been reported in various years. But in case of the, it may be reached up to 70 to 80% if climatic condition is rainy season is there, then particular season it may be increased up to 70 to 80%. And this anthracnose and stem introt, we know that these are major quarantine diseases. Actually, these two diseases, a lot of the consignments has been rejected. So we take a lot of care for the management of this particular disease before the send the consignment to other country. And other thing is the very important that latent infection is occur. Actually, these anthracnose, if we are talking about anthracnose, infection may be occur during the flowering season. And we can identify this particular inflorescence or fruits infected with the anthracnose until the ripening stage. So here average country-wise the losses we have got this information. In case of the developing countries, losses were near about 30 to 40%. But if we are seeing other country like Mexico, Brazil, and some developing country, losses was reducing up to the 5 to 10%. It's indicating that in developing country, post-harvest losses, I think 3 or 4 times more. And these are the challenges for us. Here we have found what is the comparable developed country and developing country. Average post-harvest losses were recorded 8 to 15% in the developed country, while in developing country it may be 20 to 40%. Less loss under the poor handling up to the 20%, while in the developing country it may be up to 40 to 50%. And other thing is that export rejection due to the disease is very low in case of the this developed country, but in developing country export rejection is very high due to this particular disease. Another thing, shelf life is very important. In case of the this developed country, shelf life of mango near about the 21 to 35 days, and in our country, our developing country, it may be only 7 to 15 days. These are the major post-harvest losses in the mango: anthracnose, stem introt, alternaria rot, aspergillus rot, rhizospus rot, and so many post-harvest diseases. But today we are talking about the management of the anthracnose diseases. As anthracnose symptom of the mango, actually this disease appear on the leaves, fruits, stem, and flowers. All these stages are very much susceptible for this particular disease. So management is creating a lot of issue for the appropriate management of anthracnose disease in case of the mango. These are my institute orchard. This tender leaf, you can see the tender leaf are highly infected with the anthracnose disease. Means tender leaf is very susceptible condition, a susceptible stage for the occurrence of the disease. During this period, management of anthracnose is very, very important. This primary infection, flowering stage, primary infection, leaf infection, latent infection. Actually, latent infection is very critical for the observation point of view. Estimate loss due to the anthracnose, actually maximum losses was recorded in the post-harvest in Uttar Pradesh, Andhra Pradesh, and Bihar, and other states also have the post-harvest losses, but maximum was reported in the three states, that is UP, Andhra Pradesh, and Bihar. What was distribution of the mango anthrax? Actually, mango anthrax throughout world, this disease prevention has been occurred, and highly prevention was reported from the South and Southeast Asia, Africa, and Latin America. And moderate infection was reported from the Middle East and North Africa. Low and negligible prevalence was reported from the temperate regions such as the European, North America, and Australia. Reason for the rejection of the mango consignment during the export. Actually, we have find out this particular data from the EPIDA. Actually, near about the 35% rejection due to the fruit fly, anthracnose, as well as the stem and rot. Direct quarantine rejection has been reported from this particular issues. After that, this highly pesticide residue. Actually, we know that in case of the pesticides, in our institute nearby, the farmer are spraying more than 6 to 7 spray within three months of the fruiting period. So rejection was this 45% due to higher pesticide used in the mango orchard, and 50% is rejection causes was the improper post-harvest handling and sanitation, and 5 to 10% incomplete, incorrect phytosanitary documentation, and 10 to 15% temperature and transit-related quality rejection. Actually, if we combine both 35% and 45%, near about 80% rejection of the consignment only due to the either pesticide as well as infestation or infection of the this fruit fly, anthracnose, as well as stem and rot. So we have developed a standard operation protocol. What is the appropriate protocol for before the export of our mango to another country? This principal diagnostic central dogma of the life. Actually, we are trying to identify. We are developing the molecular tools for the identification of this Clototrichum before the infection. So here we can see multiplex PCR assay for detection of Clototrichum grosspride is using ITS14PL pectin lyase and glutamine synthesis primer. Actually, in case of the Clototrichum grosspride, we can see 3 type of layer we can see in here, and other Clototrichum species only 2 or 1 or 4 type of the binding we have noticed. So specific characteristics 3 type of the layer if we can found, we can say this is infection of the Clototrichum grosspride in case of the mango. Actually, our institute has developed very good technology that is called METWAS technology. METWAS is nothing, it is the metabolites. Actually, due to the application of the METWAS, this we can increase the shelf life of the mango near about 40 to 45 days. Actually, we can see here METWAS plus hot water treatment. We can, if mango treated with the METWAS plus hot water treatment, shelf life may be increased up to 48 days. And after that, if the Sahari, in case of the Sahari, if we are using the METWAS hot water treatment, it may be shelf life increase in the 45 days. Another thing is the Baganpalli, we can 52 days we can increase shelf life of the mango, and in case of the Chausa, almost 52 days. These work actually work done by our institutes in the leadership of our director, sir. And this pilot shipment of the mango, actually due to the METWAS technology, we have export mangoes to 3 or 4 countries. You can see here in Baganpalli through the sea route. Actually, this METWAS technology, especially for the sea route technology, not for the this cargo technology. So in sea route, near about 30 to 40 or 45 days is required to export our community to the another country. After treating of the METWAS, it may be shelf life increasing up to the 45 days, up to the 55 days. You can see here, Baganapalli, okay, sir. 7 to 7.5 ton we have export in UK, Japan, and USA also last year. And here this we can see assessment of the secondary metabolites based on the formula CIS and MESWAC. What is the basic differences? Just we can show to here. T1, fruit after 14 days. When this fruit reach into other country, after 14 days, symptoms, some blackish spot we have noticed after 14 days. But if we are treating metwas plus ethylene absorbent and gamma radiation C, after 14 days, greenish color is there. It means increasing synthesis pathway is inhibit. That is ethylene synthesis that is responsible for the ripening of the mango fruit. It may be inhibit through the metwas technology. And due to this reason, we are exporting. And other thing is the metwas is very much important. This this TSS value is high. Under the if we treated mango fruit with that metwas, TSS value may be high, and this firmness is also low, and firmness is also high. Acidity is low. This is the positive factor to our mango in mango quality management. And management is also important, but I will not go to details. These are basic things. But one thing is very important. Canopy management is very important. Why is it very important? Actually, before last 2 years before, this our forest department not allowed to cut the single branch of the mango. Due to effort of my institutes, this state government has passed one cause. Farmer can cut your mango up to the 17 feet, not less than 17 feet. So canopy management very important. Due to this adhyades, we are using the drone technology for the management of insect pests and diseases also. And these are simple management practices. And another thing important is the hot water treatment. In case of anthracnose management, hot water treatment is very important. Our institute, our division has been exclusively work for these particular issues. Actually, before that, recommended technology of 52 degrees centigrade for 10 minutes. But if you have the huge amount of the mango, it will take too much time. So we have standardized 70 degrees centigrade up to 30 seconds. It's same working as the 52 degrees centigrade at 10 minutes. So if you have a lot of mango, then you can go from the 70 degrees centigrade for 30 seconds. And this is last one quotation is reducing post-harvest losses is equivalent to increasing food production without planting a single additional tree. How much important post-harvest management, we can see this particular line. So with these thank you very much. I would like to thank the organizer. He provided me opportunity to invite our talk here. Thank you very much, sir.
Thank you very much, Dr. Deepak Singh. I think you will also come out with a solution for mango malformation in the near future. Mango malformation is a complex disease that is spoiling the whole of mango industry. Thank you very much for nice presentation. And now there is a request from the organizers that to cooperate with the time saving, that Dr. H.B. Singh is waiting to conduct panel discussion. Therefore, I request Dr. Debasis, Dr. Chalam, and also Dr. Slavika from Italy to confine themselves to 10 minutes, if possible. But nevertheless, I think they will manage. So now Dr. Debasis Chakrabarti from CSIR National Botanical Research, she is going to speak on arsenic in rice. Approaches in reducing the arsenic level in grains. 10 minutes, sir. Sorry for that.
Yeah, no problem. Respected chair and co-chair and dignitaries, and I see Dr. Lavanya, who is a very senior person. So today I will be talking about arsenic, a little bit different. As you know that this is the person actually, Deepanga Chakravarti, who actually first discovered that the arsenic problem is there in Ganga Meghna Brahmaputra plain. And after that, it has been noted that irrigation of arsenic water in the field actually leads to accumulation of grain arsenic in the rice, and not only in rice, and there were several vegetables. So today I am working for last 20 years. So today I will be talking about two different approaches how to reduce arsenic in rice grains. One is through transgenics, which is not acceptable in India, but we have a technology. And another one is the genome editing. Sorry, because I don't have time, so I don't want to show you the slide, because this is the person in a village who is having so much of extended family. Now only one member remains. All people died because of the cancer due to arsenic. So that's what he actually suggested during our visit. So what we did, actually, arsenic exists in two forms, inorganic and organic. Inorganic form is toxic, organic form is not toxic. Okay? So what we understand is the lower organism have a capacity to convert inorganic arsenic into organic arsenic. So what we did here, what is the, this is the, oh yes. So what we did is we used to have screened the rhizospheric arsenic contaminated soil and then identified a fungi which is named as the Westerdekelia orientica and identified a gene which is named as a arsenic methyl transferase. Arsenic methyl transferase means transfer of methyl group to the inorganic arsenic. That makes it organic arsenic, which is actually non-toxic. And if the two groups or three groups of methyl is actually incorporated into the inorganic arsenic, it becomes trimethyl arsine, which is a volatile compound. Okay? So what we did is that we identified that gene and then we expressed it in yeast, and we found here, as you can see here in the ICP-MS data, that this arsenite is converted into several dimethyl arsenic or monomethyl arsenic methylated form. And also you can find the below things here that you can find. Where is the? No, not working. Come. What is this? So you can find a lot of volatile arsenicals are also you can find in the red circle. We also purified that proteins, and then we try to understand that whether their kinetics is better than the rat arsenic methyl transferase or not. And we find that yes, the Km value is 0.19, whereas the rat liver arsenic methyl transferase is 0.25. So lower the Km value, the better the enzyme activity substrate. So our gene is having a very good Km value. So we also did the in vitro assay, and in vitro assay also it actually converts arsenite into the methylated form, dimethyl arsenic or monomethyl arsenic. As you can see, if we give the monomethyl arsenic, it immediately converted to the dimethyl arsenic. So it's a very important gene. And this important gene we transferred into Arabidopsis. The Arabidopsis is the basic model system. So in Arabidopsis, you can find that the overexpression in Arabidopsis gives a better phenotype. As you can see, there is a reduction of feed arsenic in around 31 to 36%. So that is a very important data. We also transferred it into rice. So rice transgenic is also having, as you can see here, I don't know what is happening here. So this is a yellowing, this is non-transgenic, and this is a transgenic when we give the arsenic treatment. And as you can see here in the downstairs, that you can find that whether the arsenic grown in the irrigated water, you can find the non-transgenic panicle and the transgenic panicle. So you can find it better here. Also, the arsenic content is reduced to 50% in the grain level, as well as the volatile arsenic is quite higher here. As you can see, the volatile arsenic content is very higher. However, this technology is available, but I cannot go into the field trials because of the several reasons. I hope that the taboo will be removed soon. So that's why we go into the another technology that is the genome editing technology, which is one of my student Pooja is working actually. And we find through the transcriptome analysis that one transporter, that is an inositol transporter, is actually playing a very important role for arsenic transport. So we try to see its expression in the several different stages of rise. And what we find that during the treatment of arsenic, the flag leaf seed and the lemma, legume, the content is very higher. The gene expression is very higher. So what we did is we just express it in the mutant of yeast, and we find that it is actually uptaking arsenic. And when it uptake, the yeast obviously became the sensitive. So it's sensitive, and also we can find that it accumulates higher amount of arsenic. That indicates that yes, this transporter is actually uptaking the arsenic. Now, what we did is we did the genome edited Cas/Cas9, and we obtained two genome edited line, that is 52 and 72, where the transmembrane helix has gone because there is a mutation, targeted mutation. Because you know in CRISPR-Cas there is a targeted mutation. So we did targeted mutation, and here you can see in this slide that this is the control and this is the treatment with arsenic. There is a huge difference of the root and all these things, phenotypic character also. And you can find that these knockout lines, they doesn't have that much of yield penalty as compared to the wild type. So this genes doesn't give the yield penalty, but it reduces the arsenic, not only in phloem, but also in the arsenic content in the grain. 60-70% reduction of arsenic in grain. So this transporter, if you mutate it, you can have the plants with reduced arsenic about 70%. So we also try to identify that where this transporter is located. This transporter is located in the membrane, and also we try to find out where it is actually localized. We found that it is the phloem specific, the muscular bundle specific, and also you can see in the down right panel that it ligule, and then it is expressed in the ligule. So that where the actually transport occurs. So when you mutated this gene, they cannot transport arsenic inside the grain. So thereby reducing the grain arsenic into the grain, and it actually accumulates arsenic in the root, not even in the shoot. So we did also an analysis that how actually this gene is actually regulated. Yeah, I just complete. So these three genes, we did some yeast one hybrid. We find some of the genes that actually interact with this particular, actually regulating this expression. I'm not going. This is the promoter region where actually those genes bind and regulate. And this is the phytate content is also measured. We have the essential element also. And this is all about the summarized amount of these things. I'm sorry, I don't have time. So thank you for.
Thank you, Dr. Debasi Chakravarti. Very interesting observation and very interesting paper. Hope that in India we will have rice without arsenic in near future. For your efforts, thank you. Now I call upon Dr. V. Seliachalam, Principal Scientist and Head, Division of Plant Quarantine, ICAR, National Bureau of Plant Genetic Resources, New Delhi, to talk on role of genomics for detection of plant pathogens in quarantine. Dr. Chalam, Madam. Quarantine is a major issue in India. Hope that she will finish in 10 minutes.
Even you can take it as presented, sir.
Okay, sorry.
Good evening, everyone. Respected Chair, Dr. Manoharachari sir, and co-chair Umesha, and all the distinguished delegates. I think
Online.
there are no audience here.
Italy.
At least when we conduct the training, we have 25, 30.
कितने minutes रखा क्यों?
All the single audience, they may not require this. Anyway, the topic is role of genomics in detection of plant pathogens in quarantine.
Yes, madam.
Yeah. Like this slide I kept because you agree with me that because of the trade with the commercial material, because of the high volume, there is a high risk. That is actually what the general public and scientists also perceive. And it is both for the imported export and germplasm exchange. These are small scale seed lots for research and development purpose. During my recent visit to Jakarta for phytosanitary expert consultation, Vietnam said they consider the germplasm as low risk. But during my further slides, you will see that in germplasm, we have intercepted more than 100 pests, including fungi, bacteria, viruses, nematodes, weeds. So the germplasm also has the high risk. And these are the examples all the pathologists know, especially the famous Irish famine where 10 million people died and 10 million people migrated to USA. That is because the pathogen has migrated from Central America to Ireland. And all these pests, pathogens, they don't need the passport visa like us. They have a free transboundary movement. And these are the recently introduced pests, and most of them are insects. Only the tomato brown rugose fruit virus in 2023. Then you might be wondering, then what is it quarantine doing? We still have 1,200 pests not reported from India, and they are regulated as per the Plant Quarantine Order 2003. And to ensure biosecurity, what we need is a regulation. We need diagnostics. Why I'm saying is, if I don't want bean pod mottle virus to come into the country, the plant quarantine orders should specify that we don't want that virus. Then only it has value. And of course, the phytosanitary treatments and capacity development.
Okay.
And the international regulations by the international standards for phytosanitary measures, there are 47 of them. One is repealed. And ISPM 27 talks about the diagnostics. And this particular slide is for Xylella fastidiosa, not reported from India. And then all the protocols are available. And then India is the only country, it is a two-tier system. Every other country, if you go to USA, Australia, France, any other country, the quarantine system is a single system, both for commercial and germplasm. But in India, it is commercial is dealt by Directorate of Plant Protection, Quarantine and Storage, and germplasm is dealt by NBPGR. And it has its own advantages. And then I'm sure that my private industry colleagues will agree with me that the facilitation of germplasm is done especially for R&D purpose effectively. And Directorate of Plant Protection Quarantine, they have 73 PQ stations. And my structure of presentation, what we do in quarantine and what is the role of genomics. And ICR NBPGR is the nodal agency. As I said, we issue the import permit, give the quarantine clearance, and also issue the phytosanitary certificate for exports. And also we do the pest risk analysis. Like in the afternoon session, we had a speaker from Kenya. The same way we also do. And during the last 5 years, we have quarantined 7.5 lakhs germplasm for import, and almost 2 lakhs germplasm was exported, and 294 phytosanitary certificates were issued. And during the last 5 years, more than 100 pests were intercepted, including 41 pests not reported from India. And this is how the entire quarantine procedure is done. All the scientists, technical officers visually examine, and then the samples goes for testing to individual labs. Like this is the joint inspection by all the scientists. Whatever it is, seed or cutting or rooted plants or tissue culture plants, we examine. And for me, biosecurity is a holistic approach. If I only talk about pathogens, it has no meaning. It should be for insects, pathogens, nematodes, and weeds. We detect the insects using soft X-rays. Absolutely, they look healthy, but when we expose them, you can see the dots here. Those dots are infested ones. And these are the red color ones, representative ones. And for detection and identification of fungi and bacteria, we use both conventional and molecular techniques. And the same way for the nematodes also, we use both conventional microscopic identification and molecular techniques. And these are the weeds which were intercepted in large number during the last 5 years. These are the ones not reported from India. And the legumes, if you are importing soya bean, mung bean, urad bean, faba bean, pea, all of them are grown in post-entry quarantine greenhouses. And all the symptomatic and representative healthy samples are analyzed for the viruses. You may be thinking, what happens to the rest of us? The rest of us are grown in certified post-entry quarantine facilities, certified by the head quarantine and OIC Regional Station, Hyderabad. And during the last 5 years, like around 3 lakh samples were inspected for 336 consignments. And this is one I have kept, the recent one, how actually the post-entry quarantine, all the infected ones are uprooted and then incinerated. You can see them here. And then the infected ones are tested in the lab. And during the last 2 decades, we have intercepted 52 viruses, out of which 25 are not reported from India, using all the electron microscopy, ELISA, and PCR techniques. And even the transgenics, we do the quarantine in the containment facility where we have high efficiency particulate air filters. And more than 16,000 samples are quarantined. And transgenics doesn't mean that they come very healthy. No, we have intercepted Peronospora manchurica. These are the 5 viruses not reported from India in transgenic soybean were intercepted. And also, when you get fever, what do? You take paracetamol. Like that, when we find the infested one, we give all these phytosanitary treatments to kill the pathogens, to kill the insects. And these are some of the techniques I have mentioned. And after all this, we follow the standard operating procedures and uproot and dispose the material which is grown in our facility and also at the indenter side. The right side one is at the indenter side. And the role of genomics, it helps in especially like if Dr. Ravi Ketrapal is here, the former head of the division and now executive secretary APARE, he says the taxonomies are endangered species. So like if you don't have a good taxonomist who can identify using the microscope, then there is no other way than the genomics. And this is developed in our division. This is ITS gene sequence-based detection of Peronospora manchurica in soybean, which is not reported from India, ITS sequence. And this is the new host, Dr. Jamil is here. His group has identified this new species using the sequencing. New host record, sorry. And then Xanthomonas campestris, again a quarantine pest, which is the PCR methodology was developed in our division. And these are the viruses, I'm basically virologist, so you may see a little bit more of virology than other slides. Cherry leaf roll virus, tomato ring spot virus, bean pod mottle virus, not reported from India. And also we have the triplex PCR protocols. Instead of single PCR, in a single tube, you can detect 3 viruses at a time. And this is the quintuplex PCR, 5 viruses we detect simultaneously in soybean. And these are again a quarantine viruses. And this is the multiplex PCR for detection of 4 fungal pathogens in soybean in quarantine. This is a pathology group, Dr. Jamil, Dr. Aradhika Tripathi. And this is for multiplex PCR for both fungi and bacteria in quarantine. And these are real-time RT-PCR protocols and helicase-dependent amplification, loop-mediated isothermal amplification. I'm not going into the details. These are again developed for viruses not reported from India. And this is the reverse transcription recombinase polymerase amplification assay for detection of peanut mottle virus in quarantine. And this whole genomic sequencing is used for detection of seed-borne viruses. And genomic tools are also helps in tracking global movement of pathogens. And this helps in the surveillance and then monitoring programs. And in USA, FDA uses the high throughput sequencing and genome sequencing to identify and track the foodborne pathogens. And there are some examples like weed blast, which appeared in Bangladesh in 2016. There is a consortium who worked on this, and then they analyzed the sequence of 537 worldwide distributed Magnapothy, and they come to the conclusion that emergence of weed blast in Bangladesh and Zambia is caused by the B17.1 Bolivian isolate. We all know that it has been said that the weed blast was introduced due to series of grain imports from Latin American countries, and then the molecular genome sequencing has further confirmed it. And then multi-locus sequence typing, Ralstonia solanacearum, which was identified in the US and Europe. So it was introduced through the potato imports. Yeah, sir has got up, so I will conclude. And whole genome sequencing of potavirus was also found that it has migrated from South America to Europe and Europe to Africa. And ICR NBPJ does lot of training programs. And since 2000, we have conducted more than 30 training programs. And in 2015 to 2018, both quarantine and customs officials were trained on biosecurity, phytosanitary treatments, diagnostics. And the beauty of this training was we didn't call them to Delhi. We went to the airports, seaports, and then 4 of us. We went and conducted the training programs. And this is the right side one is 10 days training program. In this, we have included the private industry and also the plant quarantine officials, of course, the ICR Institute, SAS, and CG centers which were there in India. And yeah, in the recently National Plant Health Mission, all of you know that USA and Australia, they have the plant diagnostics networks, and the movement from one state to another will move as per the requirement. For example, Iowa State to Oklahoma, the material moves as per the requirement of the Oklahoma. But here from Andhra to Karnataka, we can just take. And we have proposed this National Plant-Based Diagnostic Network with other ICR institutes, plant quarantine stations. And we are expecting that the funds will be released for this, and then we will be able to begin. Over, sir. And then we have estimated, like, if these pests have been introduced, we could have, they could have caused the probable yield losses of 1,519 million rupees. And so for pest diagnosis, use the best tool available for the job. They should be reliable, repeatable, and reproducible. And during this, one of the conferences in Brazil, they have taken us to the fields and then they showed us the material, which is one of the Xylella fastidiosa, which is not reported from India. Anybody would get tempted to get the material to India, but I have not brought. And when you visit abroad, please don't bring any material, any seed which looks healthy, but they have the pests. So I conclude here. Yeah, these are my scientists, technical officers, triple S from both Delhi and Hyderabad. You can see Dr. Jamil Akhtar here. And I acknowledge for the funding. Thank you for your attention.
Thank you, Dr. Chelam, Madam. You have been source of inspiration for many youngsters, and Indian agriculture will be safe and plant quarantines will be strengthened under your leadership. Hope that we will be having good agriculture, and healthy nutrition. Thank you very much.
Thank you, sir. Thank you for not stopping me.
Now, last one. I think already we have lost patience, but nevertheless, we cannot ignore a foreigner, Dr. Slavika. She is from Italy. She is going to give recorded lecture. Sorry to Dr. H.B. Singh for delayed things. The things are getting delayed. Sorry for that. Ab kar do bhaiya? Dr. Slavika. Last.
Good morning, everyone. I'm really happy to participate in the ISMPP 5th International Conference at Amity University in India. To begin, I would like to express my sincere gratitude to the organizing committee for the invitation and for providing this vital platform to discuss pathogens, plant health, and food security in the context of climate change.
For people like me.
My name is Lalica Matic. I'm professor of plant pathology at University of Palermo, Italy, and research associate at the Institute of Sustainable Plant Protection in Turin, Italy. Today, I will share with you our recent work regarding in-field diagnostics of plant pathogens and innovative sensor technologies for precision agriculture. As we face increasing pressure to protect our crops sustainably, the ability to detect threats directly in the field quickly and accurately is becoming a cornerstone of modern agriculture. As you can see here, precision agriculture relies on a massive network of data, from satellites and drones to soil sensors. However, there is often a missing link in this ecosystem, the ability to detect specific pathogens in real time, directly in the field before they spread. While drones shows up where a crop is stressed, they don't always tell us why the crops are stressed. That brings us to the need for the specific in-field diagnostic tools I'll be discussing today. Through the application of available knowledge, the detection of plant pathogen in general is based on the use of traditional, destructive, label-dependent, laborious, and expensive molecular methods. The emergence of new disease epidemics is highly probable, and particularly due to the ongoing climate change, the globalization of the seed market, and changes in agricultural management practices. Thus, there is a need to develop sustainable, farmer-friendly, quick and low-cost pathogen diagnostic strategies that are in line with the objectives of the strategic emerging policies of sustainability and protection of natural resources. Therefore, new diagnostic tools, ideally suitable for field testing of plants by untrained personnel, using friendly and inexpensive equipment and providing results in a short time, would be extremely important for preventing disease spread. And that means for rapidly diagnostic plant stresses before the appearance of visible symptoms, we are testing some of these techniques that are LAMP, near spectroscopy, Raman spectroscopy, and electronic nose. These techniques for development of precision agriculture for plant stress and pathogen detection need to have in common characteristics. They should be in-field portable and deployable instruments. They should be continuous real-time monitor the plant health, and they should have actionable management based on anomalies in the real-time directly in the field. First of this technique is loop-mediated isothermal amplification technique, or LAMP. LAMP technique is one of the innovative techniques that is increasingly used as alternative to conventional PCR molecular technique. It is technique that operates always at single temperature around 65 degrees Celsius, takes only about 30 minutes for the detection of plant pathogens, and requires at least 4 primers. Here we use the LAMP technique to try to detect plant pathogen that is causing powdery mildew and hazelnut. We designed lamp primers in 2 different genes, ITS common fungal marker and RPB2 alternative fungal marker. The better results we obtained with the ITS primers, which amplify the target region of powdery mildew in less than half an hour, both in the lab starting from DNA fungal extract or also directly in the field starting from crude plant extract containing the powdery mildew lesions. So this technique work in the both conditions, in the laboratory, in the field, approximately at the same time for detection. This is how the lamp assay is working. We have the infected plant leaf, or we have infected plant fruit. Then we take this plant material, put it in the experimental tube with the tungsten bead and extraction buffer, shake it manually for a few minutes. After that, put it with the isothermal mix in the lamp machine. Then after that, we are running the lamp reaction directly on the cell phone, and we see the results of amplification in the less of half an hour. When we have amplification curve, we have the detection. When we don't have it, that means our samples and plants are healthy. The lamp assay
We had
that
10
was optimized
presentations
to work with
in
crude
this
plant
particular
extracts
session,
by manually
and
heating the leaf.
all the presentations are highly productive, and a lot of original data has been included in this. Now I would like to thank the ISMPP and organizers for having given us an opportunity to chair the session, and I give it to this mic to Professor Umesha. Professor Umesha is
Thank you very much, sir. In this session, we had 10 presentations. Out of that, 2 are online. Rest of the presentations are offline. Subjects covered from various disciplines, all including plant pathology and agronomy and the breeding. So with this, we have come to an end of this particular session. I, on behalf of all of you, thank ISMPP and also the Amity University organizers for having provided us the opportunity to serve as chair and the co-chair. Thank you very much, madam. Thank you.
Thank you, audience, for such a patient listening. And now we are coming to the last program of this today, that is panel discussion. First of all, I would like to invite the discussant, Dr. Abhilasha Tripathi. Ma'am, you are here? Okay. Dr. Pratibha Singh, please come on the stage, ma'am. Dr. Manzoor H. Thar is not here. Dr. R.G. Agarwal, not here. Dr. Sachin Tomar, Shri Arvind Kumar Gopal, Shri Jamed Akhtar, Shri Shalil Singhal. मैम एक दो वहां पे मैं शालिनी मैम किसी और को बोल रही थी, उनको भी बुलवा लीजिए. आप नाम बताइए, मैं बताऊं. Dr. D.S. Mishra is here. Please be on the stage. अच्छा. Okay. Dr. H.B. Singh, session chair. Dr. H.B. Singh is here. Co-chair Dr. V. Cheleya. Celia Chalam. Dr. V. Celia Chalam, co-chair, please do come. Report. Dr. Ashish Kumar Gupta. Dr. Ashish Kumar Gupta. Anyone else? Dr. Rakesh Jaimal. Okay. I hope. Dr. Nimisha, please be here as a reporter. Dr. Vishal Somvanshi. Dr. Vishal Somvanshi. Yes, please do come up.
Anyone left?
Dr. Vishal. So without taking much time, I ask chair and co-chair to please start the session. Who is the chair? Sir, please give permission to start. I am the moderator.
Yeah, you can give me the list of people who is
Sir, I'll be asking questions.
I will be asking questions.
Yeah.
Okay, thank you. So I think we are running behind the schedule, so we can quickly move on to the business. So friends, you all have witnessed for the last 2 days, we have a marathon discussion on plant disease and their management using different kind of modes, chemical control, biological control, and other methods. So in this plenary, we will have discussion, and you are free to respond to the questions raised by the individuals. That's what exactly we want. So I think we can
Thanks,
ask,
sir.
we can ask the moderator to please carry on.
Yeah, thank you, sir. So any of the panelists can take this question. I am not directing anyone that this question is for you or this question. Panelists are free to take the question. How do biosecurity risk impact international trade flow and supply chains? We have recently seen number of threats. COVID was one of them.
We have not put this.
So, ma'am, you may answer.
I think let one of the
Ma'am,
panelists
will
answer.
You answer, ma'am, please.
Yeah, I mean, I can just share one example, which I think in the morning I shared with the wheat blast disease. So it was related to the trade. Directly, there were like intercontinental or intracontinental movements.
Yeah.
So intercontinental were basically trade due to trade in Bangladesh. So that's one example. So how it is impacted? Actually, it's not only, it also, we realize that it's going to be also affecting food security because it's the Bangladesh region is very near to Indian wheat growing region. So it's not, so the question you ask is like how it affects.
Yeah.
So that's to the extent of food security. So
Yeah, thank you, ma'am. And I hope we all agree with it, because if the Iran war lasts for more than 2 months or maybe more, then food security become a threat to most of the nations. So now my next question is, should countries impose strict biosecurity checks on import-export, even at the cost of trade efficiency? Because whenever we put some stringent restrictions, it hamper our food security and efficiency. So, yeah, ma'am, sure.
See, this is as per the WTO. Whoever is the member of the WTO, it is not that each country, nobody can put any extra stringent. No, this is as per the pest risk analysis. Each NPPO is National Plant Protection Organization. For us, Directorate of Plant Protection, Quarantine and Storage, and for USA, it is FS. So every country will have the regulated pest list and science-based. It is not that I don't, because I don't have a right relation with Pakistan, so I cannot say that I don't want to. It is science-based pest risk analysis. Now, after 1995, it is entirely science-based pest risk analysis. Nobody can put extra conditions or less conditions.
Thank you, ma'am. Now, a quick question. What entrepreneurial opportunities are emerging in biosecurity? For example, diagnostics, surveillance, biotech. There are a number of opportunities emerging for entrepreneurs. So how do you visualize it? Yes, sir. Please, Shyam.
Madam, as far as your question is concerned, in fact, there's no direct entrepreneurship type of thing, but there is an opportunity to develop a skill, especially in the field of diagnostics, because that is the field where entrepreneurship can be developed. Right now, we have few marketable devices for diagnostics of field level pests, because surveillance is one of the important activity for maintaining the biosecurity of the nation. So this is the field where we have to work.
As per the national standards for phytosanitary measures, NSPM 23, DPPKS is accrediting the phytosanitary service agencies, PSA, and phytosanitary requirement providers. If you just Google it, NSPM 23, you have all the information. And only thing is, if a private company like Bayer, if they want to have their export promotion, they can apply for these PSAs, and then but their staff should be trained at a National Institute for Plant Health Management. It is a 3 weeks or 1 month training. So once they are trained, for your export promotion, you can apply for PSA, and some of the companies are already recognized, and Syngenta has applied, I think.
Thank you, ma'am. Now, related to it, another question is, what are the biggest regulatory hurdles that we face when we have to have a biosecurity startup? You can give the reference of the country like India or maybe any other country where they are starting the startups, with biosecurity startups.
I think Dr. Dev Mishra should tell what are your hurdles for Indian bioplant quarantine system.
Yeah, that is very good question, madam. One thing is that, as madam was mentioning, to start a startup on such kind of analysis, we need to have really clear understanding what is required, means in which country we are trying to send our material and all those. And then there is, means when a organization looks for the opportunity, they see that what kind of market potential is there. So if we see the opportunity of that kind of activity to have, then they try to invest into that one. So that is what even what we are seeing within company, like for example in Bayer. So we are strengthening our diagnostic infrastructure as well in all the region, not only in Asia, but in other region also, because we are facing a lot of issue for new pathogen that is entering from other region. And recent example has been, you all might be knowing in US, some of the material that was being carried by some Chinese nationals, they got held up at the airport. And because that was having some pathogen that was under quarantine list. So that is important for a company as well as the whole industry, as well as the country, means country as a whole as well. So those are the area we look into, and then we try to put that, yes, this is the area we need to invest more so that we can make ourselves safe when we are sending our material to target country.
Thank you, sir. Yeah, sure. Yeah.
For example, tomato brown rugose fruit virus, it was not reported from India till 1 year, and recently it was reported in Karnataka, a single report. But then Netherlands would insist on quantitative real-time PCR to test the tomato samples, and not based on degenerate primers, the specific primers. And some of the private companies have expressed they are finding it difficult to find the labs which could test for that virus. And also, apart from that, is one hurdle the private industry is facing. since we are germplasm, we don't do the commercial material. And one more thing is, as per the ISPM 45, International Standards for Phytosanitary Measures 45, there is a provision for accreditation of labs, both private and public. India is yet to do that because the trust concept comes in India. We should acknowledge that. And if you look at the East Asian countries, Thailand has accredited private industry. Philippines has accredited the private industry. East West Seeds is accredited. And even the, not Vietnam, Laos, they are trying to do that. So when China has many accredited labs in the private industry, so that is one thing maybe government of India should think about it, to have the accredited labs both in even the public private sector.
I do agree with ma'am. You want to supplement something, ma'am? Yeah, please. Sure.
Yeah, so although I am not a biosecurity expert, but Australian industry is a leader in innovation in prevention, eradication and containment of exotic pests and pathogens, building on leading research, science in diagnostic, which Celia already mentioned, and surveillance, which Dr. Jamil has mentioned, and also pest risk analysis, which is like preparedness. So like what is the pest pathogen biology, then movement pathways, control strategies, and economic impact assessment. So these are the kind of prerequisite for having a biosecurity in industries. So thank you.
So, yeah, sure.
Germplasm, like you have the Solanum lycopersicum, but if you want to import Solanum pimpinellifolium, it is not mentioned in the PQ order, then we do the risk analysis. Lots of risk analysis, we do at least 60 to 70 pest risk analysis for the germplasm. Again, the private industry should have spoken about it. For the commercial material, pest risk analysis takes little longer, 3 months, I think. That is a hurdle as far the commercial imports is concerned.
In fact, as your question was, what is the biggest hurdle for in terms of biosecurity? I'll cite one example. In the year, Celia madam, in the year 2016-17, when we imported for the first time Jatropha, there's no literature available throughout the world. This is sometimes really a big challenge for us to identify what are the threats. If there is no literature, so it's very difficult. But while examining those samples, we got so many things. At least 2 regulatory pathogens we detected for the, we can say it is for first time because there's no information throughout the world. So these are some of the real challenges. Thank you.
Thank you, sir. Very good. Now, we all know we are talking of, as per new NEP, new education policy, and leading India for 2047, we are talking of innovations. Speed of innovation do matters, and all the entrepreneurial skill is now focusing on how to develop, how to innovate something new and come in the market, commercialize the technology and go beyond it. Now, how we can ensure about the safety and compliance with this speed of innovation? Sir, who is going to answer? Innovation in sense of biosecurity. Now, when we are talking, we are now moving a step ahead towards it. Now, it will come with a cost of safety, trust, as we were just talking about. So, what are we going to do in case of safety, then its compliance? Do we have any, blueprint? Do we have any mitigation plan? As ma'am were talking, risk management plan. We are prepared for it or we are not, as in context of India.
The quarantine biosecurity, it is a very legal thing. When you say you are developing phytosanitary treatment, for example, gamma rays, or you use the laser beam for insect infestation, it doesn't stop there. Okay, you do some research and then you publish, but for the phytosanitary treatment to be adapted, there should not be any effect on the viability of the seeds, all the seeds should be germinated and there should not be any mutation. I'm just taking example of
That's
one
what I'm
innovation.
thinking about.
Yeah,
How
yeah, yeah. Safety
misuse
measures.
and other things related to it.
Yes, yes. Safety measures
big,
are there.
big issue. It is going to come. In another 5 years, we will be talking about it.
Whatever it is, in quarantine, whatever the innovations, there is a safety measures attached.
Thank you. Sir, want to supplement something?
You talked about the trust. To me, there's no word trust in biosecurity for quarantine. Trust means if somebody give the indent for import from suppose USA, even after declaration in the phytosanitary certificate, we need to test those samples because the phytosanitary certificate speaks that samples are healthy. Okay. Even after their declaration, we many times we intercept the pathogen of quarantine concern. So there is no question of thinking on the trust point of view.
Trust, not from that angle. When I said the trust on the private industry. See, when you look at the post-entry quarantine system, post-entry quarantine greenhouses, post-entry quarantine open fields are certified in the public private sector also. There is a public-private partnership and there are many phytosanitary treatments providers are accredited by the Directorate of Plant Protection, Quarantine and package houses of private industry. So in India there is a public-private partnership, but only for the accreditation labs, I said, so far no private labs. And quarantine is not based on, you believe that the sample coming from, as Dr. Jamil said, USA will not help us. No, 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 no. If you look at all our interceptions, since we get lot of germplasm from USA, that is one reason we get lot of pests. It is not that if I am friends with Nepal, I will, no, 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 there is nothing like that.
So, since we have discussed a lot about it, what do you think? Should we have standardized policy globally or should we adopt some local policies?
There is an International Plant Protection Convention and there are international standards for phytosanitary measures on how to conduct the pest risk analysis, how to identify the pest-free areas, and what are the cold treatments you should be giving, all that. There are international standards. And each NPPO, more than 195 countries which are members of the WTO, they are supposed to have their national standards for phytosanitary measures equal to the ISPM or above the ISPM standards. So there is nothing like globalized one standard. Each, like what is the pest I don't want, it is for India. What is the pest the neighboring country like Pakistan, it is for their country. For you. So there is nothing like regulated pest list is for the each country.
So we still are not having any locally adopted policy.
Yeah. Locally adapted. India has a plant quarantine order 2003. It has 144 amendments so far. And then we have national standards for phytosanitary measures. Indian regulatory system is excellent. Okay, we have everything in place.
Thank you, ma'am. What lessons from COVID-19 pandemic should shape future policies? It's still not known from where the virus emerged. So if something of that sort happened again, what are our future readiness? Yeah. Huh? You want? Yeah, no, I'll give you that.
Yeah.
Sir.
Yeah, so for that, because we don't want to have that kind of situation again, whether it is for human health, animal or plant itself. So first thing what we see that our diagnostic system should be really very stringent. Be it introduction of like the pathogen by importing material, or even if something is evolving within country also, we should be very vigilant that yes, a pathogen is evolved in one region and it should not spread very quickly in other region as well. Yes.
Because after Iraq war,
Yes.
And one day we came to get the news that there can be a chemical war, there can be biological war,
Exactly.
there can be biological weapon being used. So these are, how are we going to be, as an Indian perspective,
Yeah.
we talk, how are we going to be, future ready?
So that is where means we need to be really strengthen our infrastructure and resources so that we are well ahead of those things that is happening in real sense. So if we can pinpoint that yes, there is something having potential to spread quickly and cause a lot of damage, so we should restrict that. At least we should take some action on that part. So that is something I believe we need to really have. And I believe, as Dr. Celia was mentioning, so there is some mission of the plant health, like a national plant health mission that is being proposed. And maybe in that sense, if we can have a centralized facility, already it is there, like in Pune, they have genome sequencing and all those. So for plants also, if we have a central facility, if something is being noticed by any scientist anywhere in India, so that can be quickly analyzed and we can get to know that what is that. So maybe that, because knowing that and quickly acting, so that is only solution.
But most of the threats are unknown.
Yes.
Again, my question is, if we know the threat, it is no more a threat. So like a pandemic or COVID-19 was a threat because we were not knowing it. So in future also we have to envisage something. If it is a threat, it is unknown. So we have to have mitigation measure, maybe to human health, maybe to plant health, or maybe to animal health in that regard.
Yeah. Yeah, so I just want to add to that, like with the agricultural trade,
Perspective,
like
yeah.
probably we also need to improve our traceability because product tracing, including live animals, to support the quality of agricultural commodities. And again, your previous question, it will depend on the agricultural commodities we are talking about.
Yes.
So it's, I mean, what Celia madam was saying, like it can't be like it will be commodity specific regulations. Yeah. So
thank you, ma'am, once again. So now coming to the last question, as we don't have much time for a big panel discussion, though the discussion is going in a very true light. What is the single most important reform needed today to align biosecurity policy with global trade and entrepreneurship for a safer and more innovative future? Yeah, sure.
I think the single most biggest reform that we want to do is simplify the bureaucratic processes so that whatever policies or thoughts or scientific evidence we want to implement, we can be implemented without a delay. And that one single thing will be more than sufficient to prepare India for any kind of future.
Yeah, in fact, see, the Indian policies are very, very stringent. And if you compare with the Australian law and the US law, Indian policies are almost parallel to these two countries. They are very strict. And therefore, our assumption is if our scientists are really honest enough to perform their work, there is no threat of any biosecurity. And we have seen in the past, UG99, how beautifully we tackled and we stopped UG99 entry in our country. There are a number of such cases where our scientists, they played a very big role and today we are safe. And even in pandemic, you see here, you have seen how India beautifully managed COVID-19. So I think there is no second thought about that yes, Indian policies are very good and we have to enforce them. That is the only thing, proper enforcement. And we are the agencies, we may call quarantine agencies. If they really perform their job, nothing to worry. We are extremely safe.
I stopped putting that in the slides because people then again ask me the question. Okay. Well, like if you go to USA or Australia, your bags are checked thoroughly. Even in Australia, the sniffer dogs come and smell it. And then If you declare in the disembarkation card, there is no fine. But if you don't declare, like all of you must be aware that there is a news like one Chinese woman, she got chicken sandwich. She bought in New Zealand and she ate half, and the other half she didn't eat in the airport and she forgot. It is there in the bag. And the Australian government has imposed almost 1.8 lakhs rupees, 3,300 Australian dollars fine. It is so stringent. Like when I went with my sisters-in-law, they wanted to carry some diabetic herbs, anti-diabetic herbs. I told them don't carry, but still they carried. So I searched and then I wrote the botanical name, everything, and then I have declared. And finally they have taken it. So we don't have the bioscanners at the airports. Like if you really want to hide something and bring, you can bring from outside India in your carry bags. So that is one thing need to be improved. And another thing is this is worldwide. For online trade, there are no restrictions anywhere. Like even in USA, there is some site, Indian seed something. I also went to the site and saw you can buy any Guntur bichi or curry leaf plants, anything you can buy through online. Even in India, during the project work, I didn't get the Vinca rosea seeds. Then my project staff said, let us try Amazon. We got it in Amazon. So the online trade, there are no international standards. That was there in my last slide, but the time was less, so I didn't talk about it. So internationally for online trade, there should be international standards for phytosanitary measures. And also in India, the bioscanners when you come from outside. But of course, India is not the only country. Wherever you go, East Asian countries, even France, I was surprised. There is no checking. Like, but India.
I will just share my own experience. Dr. KP Singh is here. I was traveling from Australia, from Melbourne to Sydney by bus. So way back in 1983. So the quarantine guys stopped the bus and started checking each and every bag. One gentleman was carrying some apples. So he was caught and immediately asked to drop those apples in a container. That type of security system they were enforcing in the 1983. In 2011, I visited US and one of my colleagues, we four from Banaras University visited US, and one of my friend by profession, he was a physiologist and vegetarian in habit. So he was carrying some spices with him, particularly, the cumin and some other grain spice like thing. And he was caught at US airport. He was detained for 4 hours. And we all three were waiting for him why he is not coming. So friends, that type of system is there in other countries. Unfortunate part is we are not enforcing that. Law is also in our country, but we are not enforcing properly. So we have to enforce the regulatory agency to do like Australians are doing and like US people are doing.
Thank you, sir. Thank you all the panelists for such a healthy discussion. Now I will ask session chair and co-chair to sum up the session.
Who's co-chair?
Chair and co-chair both. Let's sum this.
Ladies first.
Yeah, I was wondering when I got the invitation to co-chair, but we were not asked to. Usually when you go for panel discussion, we have a set of questions prepared and we were not asked to do that. Then I was wondering why the session, how the session will go on. But the organizers have really brought out questions for live discussion. Although system is in place, but when you hear it again and again, you will come to know how the biosecurity system, quarantine system works in India. And it is, I'm happy that I'm part of this panel discussion because that is my biosecurity quarantine is my first love. Thank you so much.
Yeah, thank you, Celia. I'm happy that two of our scientists They are from the same field, biosafety and biosecurity, Dr. Jamil and Madam. And we have a wonderful discussion. I was also thinking that this panel discussion will cover almost all subject. But only once I arrived here, I was told it will be on biosafety and biosecurity, which is not the area of mine. But of course, I know a little bit. So thank you so much for participating. And I wish that the young people, particularly young scientists, must be very, very, happy that this type of panel discussion has occurred in this seminar. Thank you so much. Salini is coming. So Jameen, why you put it only on biosafety and biosecurity? I didn't see the program also.
It is a biosecurity session, sir.
Okay.
It is
Yeah, we can ask the questions from the audience. Audience, yeah, see, they are ready.
Hello. Yeah, very nice discussion. Just a small question. Yes, very good learner people are here. I think we will get an answer for that. The rice dwarfing virus, as ma'am has started about the COVID-19. Now that is at our door, and it is quite well shaken us 2 times, 2023 and this year, 25, 2 seasons. So what do you think, situations like that, how we can handle them? And yes, it is again, definitely it will again come. From where it has come, what were our preparations and where we lack, and how we can prepare ourselves.
Is this virus seed-borne? See, if it is a seed-borne, you can restrict the movement of seeds from one state to another. Like golden cyst nematode of potato, this is a one success story in India. Like for a long time, that nematode is restricted to Nilagiri Hills in Tamil Nadu. And later it was found in Uttarakhand, Himachal Pradesh. Then what government of India has done, the government of India, J&K also. And government of India has brought out the notification that potato will not move from these states also. See, if it is a seed borne, then it should be under domestic quarantine.
And other part of India. So there is very no any stringent rule in the government of India. Being a nematologist, the golden nematode now tomorrow it may lead to Lucknow also. There is no system at all. Man, it is only on paper, I am telling you. It is on paper that you are publishing, you are issuing order, but nothing like that. In India, everything is free.
No, sir. Potato is implemented. I'm not saying for other crops.
I'm telling you the fact, everywhere I have gone, I'm the nematologist working out 42 years. And I have seen it. There is no any restriction for the golden system. Why? Because Ludhiana, Himachal, not Himachal. It is equally Uttar Pradesh. Very true. There is no restriction.
So Dr. Pandey, can I add, you see, these microbes, particularly the nematodes, they don't have any problem. They may move from one part to other part. There are a number of examples. But the law says that if the area is infested, you should stop it by quarantine, internal quarantine. Okay. And we have a successful example of Wart disease, how Wart has been checked. Otherwise, this disease was reported 50, 60 years ago, but from that Darjeeling hill, it never moved to other part of the country. So if the problem is serious, one can take the rescue, like you have to enforce the law.
Then in last symposium, I told, who's enforcing? Nobody is enforcing. I have given 6 complaints. Last symposium, I told everyone, who's enforcing?
Yeah.
Only on paper. Nothing is.
So whom you complained, Dr. Pandey? Director Plant Protection and Quarantine, Government of India is the regulator, so you should complain to him.
आप जाके देखो जिसके प्राइवेट वाले हैं उसका बाहर कंट्रोल हो गए 50 लाखों रुपए देते दिल्ली में then they get the registration.
No, I think Dr. Pandey, these things are recorded. We should not put on record, no? We should not, discuss non-scientific thing here. Yeah.
Yes, yes.
Yeah, I think, yeah.
Sir,
So I can I can just try to answer Pankaj's question with the respect of maybe it's best to start with the diagnostics, what is the protocol for the virus, like diagnosis. So maybe you can develop. So I don't know if about the disease at the moment, but just to start the diagnostic and then start working from there. Identification is important. So I mean, that's what I'm just giving work to you again.
yeah.
And the type of laboratory they have, seed lab, you go and see, we don't have in any governmental setup that type of lab. So we should not see the private industries as a question mark. They are well equipped. I can tell you, you visit the Syngenta lab, no governmental lab is there at par with the Syngenta lab in seed technology today. So they know the latest technology, they are doing all those things, and that is why they are doing business in more than 150 countries of the world. That's see, for registration now everything is so transparent. One has to apply to CIVRC online. You can see how many applications are there, and the technology is taken from ICR Institute or some other institution. So now data sharing is also not there. You take one letter from the respective director that we are transferring this technology to this company. You submit that letter, CI will not ask any question. So things are quite transparent now. Those days are gone now. Yes, sir.
Sir, in continuation to previous question, I have I need some clarification. Why we are dependent for the import of the seed or planting material always in huge numbers? Because it is not possible to test each and every seed or consignments. So where there is a threat of import of the pest and diseases. So say, for example, why we are not looking after the replacement or alternatives? Say if we are dependent on the import of the seed material or planting material, why we are not multiplying in the country? Say if small consignment we are bringing and then we can multiply here after testing and then we can provide to the people who need it. That's one thing what I can say. Second is there is obviously some problem. Just Dr. Saab has said that some virus has come in this. I think most of the material in domestic quarantine, which is the reason for the failure of the domestic quarantine, is the ACRIP trials. You see, when material goes through ICR in ACRIP trials from one state to another state, so a lot of material is infected. You cannot stop it. And I have personal experiences in case of vegetables. A lot of viruses are coming in the ACRIP trials, which are not present in those states. So this is the biggest problem where you need to check. So if any material which is being put into the ACRIP trial, that should be tested strictly then only it should be sent for the active trials, what I have seen. So,
Yeah, that's,
yes, that's correct.
but you see, the exchange of seed material is going on and it will continue because we have international institutes in our country, like ICRISA. They have the world germplasm of 5 different crop. So their mandate is to, examine the different genotypes from all over the world and develop new genotype. So that way, I think we cannot stop exchanging the seed material from any part of the globe, but you have to see that the seed coming to our country is free from pest and diseases.
Yeah, that's why, sir, because when material comes to NVPGR, ma'am has shown in many slides, they have reported n number of pathogens from the countries which they don't know. Even they don't know that their material is infected. Similarly, some new reports. But what I mean to say, exchange we cannot stop, but we can reduce down the exchange of material so that we can test it 100%. Then we can multiply it. I think that is the one possibility. Second thing is, sir, when we are just some example you quoted regarding the apple. So then they had to throw it in the dustbin. Now you see how much apple and other dry fruits are in the market. What is the strategy with us to test them whether they are carrying some infecting microbes like insects, pests? Where we don't have, I don't think, I fail to see. You see in market walnuts, almond, or other dry fruits of any kind. So lot of pests are being pumped in the country as a gift. And it is not possible for any agency to test each and every fruit or material.
I was reading a paper, not only the consignment, even the ships are carrying pest and diseases.
Yes.
The ships coming out from, different countries. So that is a natural phenomenon. That is a natural
But
phenomenon.
some strategy should be there. I am not saying that that's obviously natural, but
Strategy
maybe in
is
future.
there, rule is there, but unfortunate part is we have limitation. Now, how many samples, NBPGR can analyze? Tell me. How many scientists are
No,
there?
that's correct, sir. They can.
So that is the biggest problem. Countries of this size, having only few scientists in NBPGR, how many germplasm they will handle?
Every germplasm we test. What sir is saying is for the commercial material, he is not indicating, but I could make out what he is saying.
In addition,
For example, a lot of apple material is coming into Himachal Pradesh, Uttarakhand, even Jammu, and there was a post-entry quarantine facilities were made, and I was part of that committee, and we have certified those facilities. But when the government takes the decision that they want to import lot of material, so there is no role of scientists here. When this project came also, we said, why are we importing lakhs and lakhs of material? We can as well multiply here also, but we don't know. Like there are other things are also involved in this sometimes. That is not in our control.
Before calling off, I would like to call Dr. C. Manolacharya of the So both of you are in. Dr. H.B. Singh, session chair, please come up.
Where is Salvi, madam? Okay, I was looking for you. Thank you, program, but
Thank you for the research. No, I haven't disappeared. I was there in the surgery. Rakesh, sir, Dr. Rakesh, please sit down. आज तक सुबह से बड़ी समस्या है। आज तक सुबह से मेरा नाम Dr. Ashish Gupta, please.
सब लोग खड़े रहेंगे अभी
Last but not least, Dr. Jamila. Sir, all the panelists, please come for a quick photograph. Thank you, everyone. Thank you so much. Thank you so much, sir. मेरा फोन आ गया